Greetings to one and all. On behalf of Ayurved Seva Sangh's Ayurved Mahavidyalaya Nashik, Maharashtra, I welcome you all in today's national webinar on Update Ayurveda, organized by Rasashastra Department. I am glad to share with you all that this is our 8th national webinar. We could arrange this webinar successfully under the able guidance of President of our institution, Dr. Ambadas Kulkarni Sir, Secretary, Dr. Abhay Kulkarni Sir, Vice President Dr. Ashutosh Yardi Sir, Joint Secretary Dr. Rajni Gokhale Ma'am, and Chairman of College Committee Dr. Abhay Sukatme Sir. A warm welcome to all these dignitaries. For inaugural ceremony, I also welcome individuals on the dais, Dr. S.L. Dasri Sir, Principal of Ayurved Mahavidyalaya Nashik, Dr. L.G. Joshi Sir, Vice Principal, and Dr. Vinay Sunambekar Sir, Superintendent of Arogyashala Rugnalaya Nashik. We are delighted to offer the most hospitable welcome to our eminent speakers of today's webinar, respected Dr. S.S. Savrikar Sir, Dr. B. Ravishankar Sir, and Dr. Ravindra Singh Sir. It gives me immense warmth and great pleasure to grace all of your presence in this national webinar. Before we begin with the webinar, let us take blessings from Almighty Lord Dhanvantari and pray for the success of this webinar. I will invite Dr. Japa Fadke for the prayer.
नमामि धन्वन्तरिमादिदेवं सुरासुरैवन्दितपादपद्मं लोके जरारुभयमृत्युनाशनं धातारमीशं विविधौषधीनां शङ्खं चक्रं जलौकां ददत अमृतघटम् चारुदोर्भिश्चतुर्भिः सूक्ष्म स्वच्छातिरुद्यांशुकपरिविलसन् मौलिमं भोजनेत्रं कालाम्बोधोज्ज्वलाङ्गं कटितटविलसत् चारुपीताम्बराढ्यं वन्दे धन्वन्तरिं तं निखिलगदवन प्रौढदावाग्निलीलं प्रौढदावाग्निलीलम्.
Thank you all. On this auspicious occasion, I take opportunity to introduce before you all our Mother Institute, Ayurveda Seva Sangha, which has been working for the propagation of Ayurveda since 1924. This selfless service to mankind is being carried out through 5 organizations which are run under one roof in our campus. Ayurved Mahavidyalaya is our college where UG, PG, PhD fellowship courses are run. Arogyashala Rugnalaya is our 100-bedded, well-equipped hospital with facilities of ICU, dialysis, etc. Aushadhi Bhavan is our manufacturing unit of Ayurvedic medicines. Sanshodhan Vibhag is the research department where many clinical trials are conducted. And Ayurved Patrika, that is our national scientific research, national scientific journal published. Few words about our mother institution amongst the most esteemed personalities who are present here today for webinar. The theme of today's webinar is Update Ayurveda. Now I call upon Dr. Shishir Pandey Sir, HOD of Rasashastra Department, to present before you all the webinar theme. At this juncture, I would like to take the opportunity to thank the organizing committee for inviting me here. I wish everyone a successful and fruitful conference. Thank you.
Good afternoon to everyone. Friends, all of us know that Ayurveda is an established ancient health science. Right from the beginning of the Veda, various new things were added at various levels in Ayurvedic literature, and today we have our own established concepts, diagnostic methods, treatment policies, knowledge of prognosis, pharmaceutics, drug delivery system in every part of the health science. Now, in 20th century, we need to think, study, document, and add various new things in between the established lines using pharmacokinetics, pharmacodynamics, pharmacovigilance without changing the basic principles of Ayurveda. For this process, first of all, we have to learn various testing or assessing methods. No doubt the methods are already established, but unlike existing health sciences, as far as medicine are concerned, we are adopting plant as a whole concept. Hence, our medicine are not having a single molecule, but having multiple molecules, achieving multiple targets at the same time. All of us know the route of administration mostly adopted in Ayurveda is oral route and per rectal route. Most of the times, we focus our line of treatment depending upon the nature of Agni, a digestive power which affects pharmacokinetics and pharmacodynamics. Taking initiative into the point, we are organizing this webinar on Update Ayurveda, and the topics chosen are testing of bioavailability of Ayurvedic medicine in animals, then stability testing of Ayurvedic medicine, and expert opinion from the authority of Rasashastra Vaishya Kalpana. Today, we are having very dynamic personalities, Dr. B. Ravi Shankar sir, Dr. Ravindraji Singh, and Dr. S.S. Saurikar sir with us to focus on the topic. Thank you.
Good afternoon, everyone. Our first speaker is Dr. Ravindra Singh sir. He has done MSc and PhD in chemistry. He has published more than 45 research articles in national and international journals. He is working in the field of natural products, pharmacopoeial work, new drug development, quality control, drug regulations. He has published 24 pharmacopoeial monographs and 80 chromatograms of drugs in PLC Atlas of API. He is having a patent of a process for detoxification of Jatropha curcas seed oil. He has completed 6 research projects related to identification and quantitative estimation of marker compounds and phytochemical evaluation, toxicity and standardization, development of pharmacopoeial standards, SOP, and shelf life of certain Ayurvedic formulations obtained from APC and CCRS. He has given major contribution in the preparation of 3 books related to quality control, drug development of Ayurvedic formulations, phytochemistry, drug standardization and quality assurance of Ayurvedic medicines. He has been awarded with life membership of Indian Chemical Society, Kolkata. Presently, Sir is working as Assistant Director at CCRS, New Delhi. I request Dr. Ravindra Singh Sir to share his views on stability testing of Ayurvedic medicines. I request Dr. Ravindra Singh Sir to share his views on stability testing of Ayurvedic medicines.
Yeah, thank you very much. Can you see the screen, ma'am?
Yes, sir.
Yeah. Okay, on the onset, I would like to thank Professor Shishir Pandeji and the principal sir and the management and the organizing committee for inviting me for this webinar. So, and dear participants, it's my pleasure to just have a talk with you about regarding the stability testing of Ayurvedic medicines. So, if you see that in recent times, a lot of discussion is being held on the stability testing of Ayurvedic drugs, and this two gadget notification made by Government of India in 2009 and 2016, and now in the gadget notification of 2016, it is mandatory for all these manufacturers to have the quality this stability testing data for their patent and proprietary item or the classical formulation. So if you see in the case of the previous notification issued in 2009, the stability test is this shelf life studies of or the shelf life of the Ayurvedic formulas, the classical formulations has been declared by Government of India, and these manufacturers, they are putting those shelf life on their labels. But now in 2016, it has been made clear by the Government of India that the the manufacturers who are having the patent and proprietary items or the classical formulations, they can claim better shelf life also, and it should be backed by the shelf life data of their product. So it means that all the manufacturers now they have to take out, or they have to work out the shelf life data of their products, and then they have to submit the data to the licensing authority for giving the license or renewal of the license. So let us see that what is the stability testing and how we have to do it. So as you all know that stability, what the stability period means, that it is a period in which the product or the medicine, it remains sound without deterioration and remain fit for use. So there are many changes in the formulation with the passage of time. For example, there may be crack in case of tablets, there may be cracking of the tablet. There may be friability, or the some churna or the powder material may come out, and then there may be a loss of color, discoloration, then growth of the microorganism in that, and then the loss of the phytoconstituent. These things may be encountered if the drug is not stored properly, or if also it is stored properly, these aging process may appear in the shelf life. So the shelf life of a product, it depends on many parameters. So for example, their ingredients, their packing materials, the environmental factors like where the drug has been stored, for example, the temperature, the relative humidity, etc. And even if this product should be saved from light, whether it is kept in a well-lit container, or it is exposed to light, or all these things. These parameters, they have an impact on the shelf life of the product. So when we used to do this shelf life studies, so it means that we are establishing that for a particular duration, that the medicine will remain suitable for consumption, and if it is stored under the optimal condition which have been told by the manufacturers in the label. So if you see that in Rule 161A, it has been made mandatory by government that all these manufacturers, they should give the label specification, and in label specification, the shelf life is one of the prominent factor. And in shelf life, in Rule 161B, it has been told that what are the shelf life of various products. So it is first of all, we should know that what is the purpose. So if you see that it is a legal provision that we should have the stability testing of our product, and we have to mention it on the label itself. And then what are the approaches, how we have to approach. Then main thing is that we have to store the sample of the same batch, because when we are conducting any shelf life studies, we have to conduct the study in same kind of batch, and at least three batches should be taken at present. And there are two type of storage conditions. One is standard storage condition, or the real time, or the long time, and one is the accelerated. I will tell you later on. And then the sample has to be, and if we are just there are two type of studies, like one is cross sectional approach and one is the normal approach. In normal approach, we have to just follow the stability. We have to just store the sample in the same batch material and standard storage. And in accelerated storage condition, we have to test them at a periodically stage. So we have two kind of things, like we have a cross-sectional approach and we have a normal approach also. So cross-sectional approach means that the samples which have been manufactured over the 5 years and spanning the 6 months, and evaluate them simultaneously. And based on the result of this shelf life, we have to discover, we have to just arrive at the shelf life of the product. So first, the design of the study. In design of the study, we have to just select the batches. We have to just find out that what kind of batches we are going to take for the shelf life. Then we have to have a specification of the testing parameters. These testing parameters may be either the pharmacopoeial standards, if it is a classical formulation, or it may be a in-house standards if it is a patent and proprietary items, and then we have to see the testing frequency also, then the storage condition, then the container closure system also. So the containers, they are also very important. For example, some of the containers may react with the pharmaceutical compound or the drug. For example, if it is the container is made up of tin, then the tin may get rusted in due course of time, and it may react with the phytoconstituent which is present in the drug. So it is also very important. So then the first of all, we have to select the batches. The formal stability studies has to be conducted on at least 3 primary batches. The primary batches should be of the same formulations, and for new product, the batches should be manufactured to a minimum of a pilot scale by the same route, using a method of manufacture and procedure that simulate the final product to be used for production batches. For example, if you see we are preparing a commercial batch of around 100 kgs or 200 kgs or 500 kgs, then we have to take at least 1/10 of that commercial batch. And suppose if the containers are too big for them to be kept for analysis or for study, then we have to simulate the same material, smaller container with the same material. And for all these things, we need shelf life chambers. So commercially, shelf life chambers are available in the market, which can control the relative humidity and the temperature in the desired degrees. So in all these things, we have to just keep these material or the the drugs into the chambers in appropriate storage conditions, and then we have to just check it on the stipulated time periods. So as I've told you that this is the definition, that the stability, it is an essential factor of quality in traditional medicine. If the drug is not stable, then we can say that it is to be used as early as possible. Suppose, for example, the swaras, they are having very low stability period, and then we have to just take it out, and then we have to just take it, and it has to be used in the patient. For example, like quat tunes also. Quat tunes we cannot store for a longer period, and then it has to be boiled. Then the quat has to be prepared, and the quat has to be taken by the patients. So, but in recent time, we have seen that a lot of people, they are in some part of the countries, they are preparing the quat tuna also in the form of decoctions, which in which the preservatives have been added. So in that cases, the aim of the what is the aim? That the aim, it is mandatory from legal point of view that we should have the shelf life of our product. And for this Ayurveda, Siddha, and Unani medicines, that also we have to we have to just work out our shelf life, and then we have to see that there should not be any microbial, there should not be any physical, chemical, or microbial deterioration in our product. So as I've told you earlier, that cross-sectional approach is there. Cross-section approach means that if the batch has been made in the last 5 years, so we can just take out some samples, and then we can also have some shelf life studies for them. So this cross-sectional approach, at least we have to take 2 batches per year to be selected, and in normal mode we have to take at least 3 batches to be selected. So if you see the container and the closure system, the stability study should be to be conducted on the dosage form packed in the container and closure proposed for marketing. Though whatever material we are proposing for drug to be marketed, then the same closure containers has to be used, and the smaller containers should be used so that it may accommodate in the shelf life chambers. So if you see the specifications, the specification, as I have told you earlier, that the specifications may be the pharmacopoeial standards, or it may be the in-house standards for our patent and proprietary items. And we have to test the specifications at the relative time. For example, at the start of the study at the 0, then we have to take out at the samples at the third month, then we have to take the sample at the 6 month, then we have to take the sample at the 9 months, and then we have to take the sample at the 12 months. And in all the cases, we have to just see that the physical, chemical, biological, and microbial attributes, what be whether there is any change or there or not, that we have to see. For just seeing all this kind of things, that thin layer chromatography is a very good technique to show that there is a degradation in the formulation. The TLC will show that whether the compound is being degraded or it is intact. For example, if you see the TLC of the product at zero day, and then 3 months, then 6 months, 9 months, and 10 months. So TLC will tell you that whether the compounds are being degraded or not, or whether your drug is being subjected to degradation or not. So specifications, maybe the chemical parameter, the physical parameters, maybe the color, the change in the color, the odor, the appearance, the shape, taste. So these are the some of the physical parameters. So we can just see that, for example, in the if it is a tablet or the vati, then we have to see that whether there is a crack or discoloration or any change in the physical attribute. So these things we have to see. And in the case of the chemical parameters, we have to see that the pH the color reaction, the pH value, the weight variation in the in case of tablets and capsules, then disintegration, density, extractive value, estimation of the marker compound, and other profiling, and the chromatographic profiling is also has to be done. Then if some assay has to be done, then assay we have to see that whether the prominent marker compound is being degraded or it is intact. So these are the testing frequency and the storage condition, the real-time shelf life study. So we have to just see that the storage condition is temperature should be 30 degree and plus minus 2 degrees centigrade, and the storage condition is the 60% relative humidity plus minus 5 degrees centigrade, and the testing frequency is the 0, 3, 6, 9, and 12 months. And for accelerated this study, we have to keep the temperature. These the accelerated studies, the short-term duration studies, and we have to just complete it in 6 month time. And we have to keep the temperature at 40 degrees centigrade and 2 degree plus minus 2 degrees centigrade, and then relative humidity at 75 with a variation of plus minus 5 degrees centigrade. So if you see that in ICH, the world has been divided into 4 climatic zones, that the zone 1 to 4, based on the temperature and relative humidity. And our India, it comes under the zone 4. And this test condition, the storage conditions have been made according to zone 4 only. So this the storage this shelf life guidelines, they has been mentioned in the API part 1 volume 1 and API part 1 volume 9 for the benefit of the stakeholders. So as a student and the teachers and other stakeholders also, you can just go through these guidelines. They are very specific and very lucidly written. So these guidelines can be read for better knowledge of the shelf life study.
सर अभी प्रेजेंटेशन दे रहे हैं तो देखते हैं कितने चलता है.
So if you see that this is the shelf one of our formulation in CCRAS. So in that we have conducted the study from 0, 3, 6, 9, and 12 months study. So if you see that there is some. If you see that some variation is there in May 2013 and August 13, if you see that this compound is being, it is just showing a lighter color here, then again it is showing the same color here, then again in February 2014 this color is gone here. So if you see, this TLC can show that there is some kind of deterioration being taking place in the product. So if you see that this band, these type of bands, they are appearing here now. So it shows that there is some deterioration in the product, and then we can just conclude that this is the shelf life of that product, and after this period, this product start deteriorating. So this is the ICH stability zone, as I have told you earlier, that the zone 1, it refers to the temperate zone, and the zone 2, it refers to the Mediterranean, the subtropical zone. The zone 3, the hot dry zone. Then zone 4, the hot, humid and tropical zone, which pertains to India also, zone 4. And zone 4B, the Asian country testing condition and the higher humidity. And if you see, these are the temperature which have been given for us. It is the zone 4, the 30 degrees centigrade and 65-degree relative humidity. And in accelerated time, then it is the humidity is 75 and the temperature is 40 degree, and the minimum duration of the study is for 6 months time. So then when we have just done our study, then we have to just evaluate that what we have to just find out of by the data we have just done. So for example, I'll just to sum it up, that in the shelf life studies, we have to just keep the around 3 batches of the material, and for every time, every this I think this time, for example, this suppose we have to just take out the sample at 0 month, then we have to keep around 3 to 5 samples of that compound in that, so that the sample should not be meager for studies. So in that cases, we have to just keep extra amount of the sample in that for 0 month, 3 months, 6 months, 9 months, and 12 months. The samples should be there, so that the analytical testing can be done for the compound, for the drug. So there are the significance change has been given by Ayurvedic Pharmacopoeial Committee, that what we have to see, that when we are evaluating, that how we can say that the compound is getting deteriorated. So if you see that A plus in this A plus or minus 20% change from the initial value, if it is initial value is there, then if the drug is analyzed for marker, if the drug is analyzed for the marker, then there may be a change, initial change from 1 to from A plus will be the plus minus 20% from the initial assay, and if in A plus, then minus 15% changes from initial value if the drug is analyzed for active component. So these are regarded to be the significant changes. So then, as we have discussed earlier, that the appearance of a new spot in the identification of the TLC when compared with the sample stored in less than 10 degrees, or completely disappearing of existing spot. So this we can just say that this is a significant change. Then the change in the physical parameters, that physicochemical parameters, they should not vary more than 25% of the initial value. And then it may be a significant change if there is a failure to meet the acceptance criteria as per the individual monograph or the specification. And then if there is a change in physical attributes like functionality, color, phase, separation, caking, hardness, etc. So if you see that to check the changes in the quality standard at various interval, it is found suitable for ascertaining the shelf life of the traditional medicine. The storage condition now it is 30 degree and 2 degree, and relative humidity is 60%. And the 10 to 12 packets of 100 grams of each sample should be packed and stored in a stability chamber, the real-time stability study, and one packet of 100 grams shall be kept under the cold storage. So it is less than 10 degrees centigrade plus minus 1 degree centigrade. So that will be it will act as a reference material. And then this reference sample has to be used for identification and the comparison of the chromatographic printing. So that may be the initial value. So this is the withdrawal time, that how we have to withdraw the samples. This real-time stability study, the sample has to be withdrawn minimum 9 times, including the initial and the final withdrawal as per the following day, the first day of the first month, then the third month, then sixth, ninth, and 12 months. And in second year, the same thing has to be just repeated. So in that, the sample should be withdrawn from each as per the time of the expiry mentioned on the label. Then the withdrawal should be within 7 days of the storage period. So that we have to be very cautious about it. And this as soon as the sample is withdrawn, it has to be sent for the analysis, and it should not be kept in the laboratory itself, because there may be some variation in the values. So this sample may be preserved properly during the analysis whenever required. Analysis should be carried out in duplicate or probably in triplicate. So if then there is a delay in this analysis, then it has to be stored or refrigerated till it is taken for the analysis. So these are the some of the stability parameters. Actually, if you see that the stability parameters, it varies with the different kind of doses form. We are having around 18-20 type of doses form, and for each doses form, this test varies. So I have taken around two doses form for comparison here. So if you see for Grita, we have to see the physical test, their appearance, color, odor, etc. And one of the foremost important test in the case of Grita and Tail is that there should not be any rancidity. The rancidity test has to be done first, because if it is rancid, then we have to just see that the stability testing has to be seen from a closer eyes. So the specific gravity, SAP value, the iron value, the acid value, the peroxide value, the total fatty matter, the chromatographic fingerprinting, the congealing point, the optical rotation, the volatile matter, assay for the marker, and the microbial load has to be carried out in the case of the Gritas. And for vertis, we have to just see the physical appearance, then the LOD at 105 degrees centigrade. Then we have to see the total ash, acid insoluble ash. All the physical chemical parameters has to be done. And this chromatographic fingerprinting is one of the important parameter in the case of shelf life study. So for test tablet also, there may be some change in the disintegration time or the hardness, then the friability, the uniformity, the total sugar, and the microbial load. If it is there, then we have to just see. So if you see that the latest gadget notification which have been issued by government on August 12, 2016, and it has been amended as Rule 161B, which says that all these manufacturers they should have their shelf life studies done for their patent and proprietary items. Even for classical formulations also, they should be done if the manufacturers want to claim for the higher shelf life value. One of the most important thing is that the more shelf life value of a drug, it means that the drug will be in the market for more time, and there will be chances of the drug to be sold. So it seems that the higher the shelf value, it will be economical in nature. So this is the some of the shelf life compiled on the basis of this gadget notification of August 2016. So if you see that the Asova Arista, it is 10 years. Then for Avleha Khan Park Good, it is 3 years. So for Dhupam Dravaka Ghret, it is 2 years. Guglu, it is 5 years. And Gutika and Vati, it is 5 years, 3 years, and 10 years respectively. Then for Kupipak Rasayan, it is 10 years. And then for Satwas, it is 2 years. Then Sharkara and Panak and Sharbat, it is 3 years. Tail, it is 3 years. And Varti, it is 2 years. So this is the shelf life for the Siddha medicines. So this is the shelf life for Unani medicine. So this is all about shelf life. So in nutshell, it can be say I can just say that the shelf life now it has become a very prominent feature of all our traditional medicine, because previously it was a general notion that the Ayurvedic drugs they have a very long shelf life, but now the shelf life has to be just it has to be proven on the basis of the experimental data, and this data has to be given to the licensing authorities for getting a proper license for all these things. And it is important that now the consumers are also getting aware about the shelf life, and then normally now people used to just check any Ayurvedic medicines with the shelf life, so they used to buy the medicine after seeing the shelf life only. So for the Ayurveda fraternity also, so this is now very good thing. And then we have to just see on just apply our brains also, that how the shelf life can be enhanced, and what are the factors which are responsible for low shelf life. For example, in the case of Taila, so Taila, they contain some free fatty acid like oleic acid. So this oleic acid, when it comes in the contact with the atmospheric oxygen, it reacts and it gives a foul smell or a rancid smell that we used to know as a rancidity of the oil. So that is nothing but a chemical reaction between oleic acid and the atmospheric oxygen, so which form a compound which gives some uh, just a disagreeable odor. And for example, for all other oils also, in most of the time that some gummy material which is present in the oil, it may get accumulated at the bottom, and it gives a ugly view to the formulation. So these things we have to just check at the time of murchna, when because in our shastras that murchnas has been given for our oils. So this murchna, they just remove all these kind of doshas. So if you see that in the case of edible oil also, there are some of kind of different kind of these parameters are there when the oil is being refined. For example, the oil it is chilled at -5 degrees centigrade to remove all this gummy material which is present in the oil, and then it is filtered. Then again it has to be treated with the hydrophosphoric acid to remove all the gums and other oleic acid. Then again it is treated with around 1% of the alkali or the sodium hydroxide solution to remove all this free fatty acids, so that it may not react with the atmospheric oxygen. And after that it has to be decolorized. This edible oil, normally the oils which are yellow in color due to the presence of the beta carotene, and we all know that beta carotene is very beneficial for us because it is a precursor of vitamin A. But in commercial oil preparations, they used to remove this edible oil industry, they used to remove this yellow color, and they used to add this vitamin E in the oil or as a preservative. But in Ayurveda, we are not allowed to use any preservative in the oils. So in that way, we have to just see that our oils, they are made as per the Shastric preparations only, so that we should have our good shelf life for that. And uh, I just want to say that we have to just follow all the standard operating procedures and to make the formulation according to the best suited SOP, so that the formulation which we are preparing for the general public or the patient, it should be of good quality and it should have a better shelf life. So with this, I just thank you all for patient hearing of this deliberation. So thank you all. Thank you.
Thank you, Dr. Ravindra Singh, sir. Thank you, sir. Thank you,
Yeah,
sir,
thank you, sir.
for very good information. Uh, Now, to start with second lecture, we are having Dr. B. Ravishankar sir with us. He obtained MSc degree in pharmacology from Mysore Medical College and PhD from Gujarat University. He is former director of SBM Center for Research in Ayurveda and Allied Sciences and worked as professor of experimental medicine. He has an experience of 33 years as Head Pharmacology Laboratory, IPGT and RA, Gujarat Ayurveda University, Jamnagar. Sir worked as Project Officer, Pharmacological Research Unit under CCRS. Sir is having vast experience as a guide and co-guide in pharmacology. He guided highest, that is more than 250 number of scholars in the country in this sector. Sir is the pioneer of pharmacological studies on Ayurvedic concepts. Sir has published 227 plus papers in national and international journals. He has been honored by Indian Drug Manufacturers Association Award in 1993, Hariom Ashram First Gold Medal for Research in 2001, Ayush Lifetime Achievement Award for Research in 2017. Presently, Sir is working as member of governing body CCRS, Government of India, and member of Scientific Advisory Committee, NIA Jaipur, and member Ayurvedic Pharmacopoeia Committee. I request Dr. Ravi Shankar sir to share his views on testing of bioavailability of Ayurvedic medicine in animals. Dr. Ravi Shankar sir.
Uh, good afternoon, everybody. First of all, I would like to thank the organizer for providing me an opportunity to interact with such a vast group of listeners. And I also apologize that due to Wi-Fi connection problem, I was not able to connect at the scheduled time of 1:30. So the topic given to me is testing of bioavailability of Ayurvedic medicines in animals. Currently, in the process of drug discovery and drug development, bioavailability measurement of a active principle or a potential chemical molecule is very, very important. Without that, marketing permission would not be given. Though it is very important for Ayurveda drugs to be accepted universally, bioavailability studies in Ayurvedic medicines have not been undertaken. This due to the fact that many complex issues are involved. The formulation view is complex. They are present in different concentration and they interact with each other to produce combinatorial active principle. They have different affinity to different entities. So complex nature. It's not easy to adapt the bioavailability for synthesis based chemicals which are which are the main source of potential.
We are experiencing some problem with V. Ravishankar sir. Stay with us. Sir will join soon. We are trying to connect with B. Ravishankar sir. Uh, please stay with us.
फिर कनेक्ट कर रहा हूँ अभी ओके ओके I was I don't know. Sorry for the disruption. That is again due to the Wi-Fi connection in instability. I was trying to provide the formal definition for bioavailability. So the bioavailability is also considered as the dose entering the systemic circulation after administration of a given dosage form. So it is considered as it is calculated on the basis of the ratio of the amount of the test drug absorbed from the test formulation in comparison to the amount absorbed from a standard formulation. So it is the bioavailability is the major determining factor for fixing the drug dosage and also determining the route of administration. It is also helpful in determining the ratio of safety and efficacy ratios. Mainly, four important physiological processes, which are given the mnemonics of ADME, that is absorption, distribution, metabolism, and excretion, studied under what we call as pharmacokinetic studies. They play very, very
presentation. Presentation. Bring it into presentation mode. उनका शेयरिंग वर्क कर रहा है राइट ठीक है
सो
अभी भी नहीं है
नहीं अभी आ गया. So, four physiological processes: absorption, distribution, metabolism, and excretion, which are studied under pharmacokinetic, they play important role in determining the bioavailability. Apart from this, age, sex, physical activity, genetic phenotype, stress, gastrointestinal disorders like achlorhydria, malabsorption syndrome, even surgical intervention, especially bariatric surgery, and interaction with other chemicals which reduce the absorption, the form of the formulation dosage which is given, which determines the disintegration and dissolution time, and also biorhythm. All these can influence ADME and through that bioavailability of a product. And from Ayurvedic perspective, it has been known that the prakriti of the person has got remarkable influences. According to a study by our Pune group, uh, people with kapha prakriti are known to have slow metabolizing abilities in comparison to pitta prakriti people who are considered as fast metabolizers, and also that's how the level of bioavailability depends upon the drug metabolizing capability of a person, depending upon the prakriti. So whenever we design any bioavailability study for Ayurvedic formulation, it is very very important to take into consideration the concept of constitution, which has except for the prakriti influence, the other factors which I have mentioned earlier, they are true only in case of synthetic drugs, which are normally used as single, very well defined molecules, whose chemical profiling is clear, and the test for identity and test for estimating the concentration, all are very well standardized. So with such product, it's not that difficult to conduct a pharmacokinetic study and determine the bioavailability related parameters. Uh, the, uh, Whenever we speak about traditional system formulations, so the situation is entirely different. As I mentioned earlier, the chemical profile is highly complex. The chemical profiling is not complete. The chemical interaction may produce new chemical combinations, and their attachment to endogenous substances, active principle present in the formulation may interact with endogenous material in a different manner, in different concentration. All these complex parameters make it a very difficult task by employing the standard pharmacokinetic study guidelines. This thing. However, some of the scientists who are working in this field, they have shown that metabolomic approach can be a better approach for determining the bioavailability. So before dwelling on the test which can be administered, which can be employed to assess absorption, distribution, very brief information about how the drugs are administered. Drugs are administered either by enteral route or parenteral route. Enteral route, sublingual, oral, rectal. I will not get into the details because it's not, it doesn't come under the scope of this presentation. So they can be given by injection like subcutaneous injection, intravenous injection, or intramuscular injection, or they may be inhaled through nebulizers, or special device may be employed to administer these drugs. The drug absorption is defined as a movement of the drug across plasma membrane uh, to enter into the site of administration uh, through bloodstream. So many processes are involved in drug absorption. Some of them are passive without any energy expenditure. Some of involve expenditure of energy, involve specialized carrier systems, which we will be dwelling in little detail after some time. And the plasma barrier, it's a very complex one, which has got both hydrophilic and hydrophobic components, mainly protein and lipids, protein of different types. So some proteins act as flex pumps, that is pumping out the chemicals from inside to outside, and there are certain influx pumps which uh, which act as carrier for influx of foreign molecules. There are channels through which protein channels through which drugs can enter. So this complex structure has to be taken into consideration. This is one of the most important determining factor in determining the bioavailability. So this absorption occurs through three categories of process. One is paracellular, that is entry of drug between the cells, either which is known as filtration or a bulk flow, or through the plasma cell membrane. The plasma cell membrane process is known as diffusion, which can be simple diffusion or carrier mediated diffusion or channel mediated diffusion, or it may be active through involvement of molecule specific transport carrier proteins, or it may be secondary to the transport with other molecules. Then we have got transcytosis. In transcytosis, phagocytosis may be the root, especially in the specialized cells like neutrophil and macrophages. Phagocytosis occurs in a non-specific manner in which the fluid in the extracellular fluid is engulfed. So any molecule which is present in that engulfed extracellular compartment will gain entry into the cell interior. Sometimes this endocytosis occurs through receptor-mediated mechanism. So specific receptors are available to promote this endocytosis. So like this is three processes: paracellular between the cells, transcellular, and transcytosis are the important process through which absorption occurs, and there are methods to assess absorption through these processes. However, this paracellular, it's not a simple passive this thing, because there are many structures between the cells. For example, three important structures are there, which are made up of proteins, different kinds of proteins. For example, there is what is known as tight junction. So tight junction fuses cell membrane of the two adjacent cells, and then we have got adherent junctions which are formed by the joining of the cytoskeleton. The first one is cell membrane joining, that is tight junction. Adherent junction, it is formed through the connection between cytoskeletal of the adjacent cells. And then we have got gap junction, in which when they the specific proteins in each of the adjacent cells, they join to form a pore through which small molecules can enter. So some of our formula, especially the piperin is known to modulate the integrity or functional dynamics of these entities. And also, as I mentioned earlier, there are influx inward carriers and outward carriers. The total dynamic equilibrium between these influx, efflux carriers, passive and active transport will have a final determining influence on the availability of drug. So transporters, there are different kinds of transporters, co-transporters, exchangers, passive transporters, water channels, ion channels, uh, proton and proton pumps, and ABC transporter carry transporters. So the exact role of each of them is not clearly defined even for synthetic drugs. So however, it seems that the consensus is that they play very important role. Uh, there are numerous transport proteins. However, some of them which are very well profile are shown in this slide. For example, these are the transport proteins given specific name depending upon the laboratory where they were characterized. So these three are involved in transport of drug from blood into the intestinal lumen, and these OATP, that is organic anion transporters. So they are involved in taking out material from intestinal lumen into the bloodstream. Like that, there are transporters which pump from the hepatic cell into the bile, and there are some which transport molecules from the intestinal lumen into the bloodstream. So these
Sorry for disturbance. There is a problem with the network. Stay with us.
So all these factors play important role in determining the absorption. Then the next component is drug metabolism. Drug metabolism can occur at different sites. It may be in lung. So it it can be through microsomal enzymes, hepatic microsomal enzymes or extrahepatic microsomal enzymes, and hepatic non-microsomal enzyme may also be involved in this drug metabolism. However, the main metabolism occurs in the smooth surface in endoplasmic reticulum located enzyme known as cytochrome P450. So I'll be giving a little more details on this a little later on. So biotransformation of drug or metabolism occurs in two phases. Phase one is known as you know uh, uh, polar molecular creation. That means if in phase one reaction, which occurs especially with cytochrome P450, mainly they are oxidative, reductive, and hydrolytic reaction in which a polar group is either created within the drug molecule or unmasked. So the drug molecule becomes more polar and easily excreted out of the body. So the majority of the phase one metabolites are generated by the CYP450. In in the oxidation reaction, nearly 11 to 12 type of oxidative reactions are involved in creation of polar groups. Then we have got three major reduction reaction: azo reduction, nitro reduction, and keto reduction. Apart from this, dehalogenation, disulfide reduction, nitro reduction, sulfur reaction also takes place with respect to some of the molecules. Then we have got hydrolysis. Hydrolysis is the process in which a molecule of water is inserted into the drug, and the drug are active, principle, and it is split into two. So in this two type of enzymes, amidases and esterases, play a major role. Cytochrome family of enzymes, they are located in the smooth surface endoplasmic reticulum. A number of different categories are there. Among them, the cytochrome P451, 2 and 3. These three families are involved in metabolizing most of the drug molecules. So the nomenclature is the CYP name is given as a general name for the class, and the number one determines the family, and 1A determines the subfamily, and A2 gives further identity. And if it is present in italics, then it represents the gene which controls that formation of that enzyme. Apart from the cytochrome P450, other enzymes are also involved in drug metabolism. Hydrolysis in plasma by esterases, alcohol and aldehyde dehydrogenases in cytosolic fraction of the liver, monoamine oxidase inhibitors in mitochondria, xanthine oxidase involved in uric acid production, and substrate-specific enzyme like tyrosine hydroxylase, beta-carboxylase. So these are some of the examples of the non-microsomal enzymes involved in biotransformation. The phase two drug metabolizing reaction involves synthetic reaction in which a polar molecule produced in the body, inside the body, is inserted into the drug molecule with the help of a cofactor, and also with the help of this substrate specific enzyme. So among them, glucuronide conjugation, sulfate conjugation, acetylation, or insertion of acetate fraction, amino acid conjugation like glycine, or glutathione conjugation, or methylation. These are the important phase 2 reactions in which endogenous molecule, polar molecule, is inserted into the drug to make it highly polar, so that it cannot, it will not be reabsorbed from the kidney and it will be excreted out. The third important component of pharmacokinetic is excretion. Excretion, many different organs are involved, but kidney is the main organ for non-volatile material. So two type of procedures are involved. One is diffusion. Diffusion, just like absorption, it is sort of a reverse to the absorption. So lipid solubility, ionization factor, the weak or strong electrolyte nature, some of the chemical attributes, they determine how much of the drug is diffused back into the circulation. However, two specialized carrier systems are available. So one is one carrier system is for organic anion transport. Another one is for organic cation transport. In this paraglycoprotein in the membranes is involved in a major manner. Apart from the paraglycoprotein, several other transport proteins are involved in renal clearance. For example, HOC2 is there. It is found to be inhibited by plant product like harmaline. Of course, it is inhibited by cimetidine, and the other group is HMAT. It mainly coordinates with the HOCT for the excretion, and cimetidine is the drug which inhibits it. And then HOCT and paraglycoprotein is modulated by a number of molecules, both synthetic and plant derived. And then we have organic anion transporters, number of active principles obtained from plants like rosmarinic acid, vagonin, rene, biacalin, ursolic acid, lithospermic acid, salvionolic acid, rosmarinic acid, aristolochic acid, many epicatechin, gallate. So each of them have got specific substrates, and they are involved, and they are supposed to be modulated. Their activity is modulated by these molecules. So what I am trying to tell is that there are several transport system available in the proximal tubule of the kidney, which play very important role in drug excretion. Not much is known about them, even how they handle the synthetic molecules. Very less is known about how they handle the active principles or active chemical components present in our medicinal plant-based formulations. But nevertheless, they play a very important role. It's very important to take their involvement into consideration whenever we determine bioavailability of any product. Another very important point to be noted here is that, so we have what are known as material which can modulate bioavailability. We have got one of the best available group of drugs in Tricatu, which has got very unique mechanism of action. Nothing is like that, nothing none of other system such drug is available. So no information, though no information is available on the involvement of Tricatu, lot of information is available about the activity profile of piperin, which is the main component of Tricatu. So it acts by multimodal mechanism. So it may activate DNA receptor binding of so that the proteins involved in increasing the bioavailability would be produced in an accelerated manner. So they may modulate cell signal transduction, so that thereby affecting the different process involved in pharmacokinetics. They may inhibit efflux pump like paraglycoprotein and other proteins, and it inhibits the drug metabolizing enzyme like cytochrome P452. Not only that, it inhibits the production of endogenous material which are used for excretion purpose like glucuronic acid. So it inhibits the enzyme which metabolizes glucuronic acid, or a factor which is involved in utilization of glucuronic acid. Not only that, it inhibits several kinds of P450 enzyme, especially C53A4. C5P3A4 is one of the most ubiquitously available and the enzyme which metabolizes a large number of chemicals. And then quercetin is present in number of our products. Red onion is the important component. It inhibits excretor, the efflux pump and metabolizing enzyme. Then genicetin, it again inhibits 3 transport proteins. And then naringin, which is the major flavonoid glycosides in the grapefruit, inhibits both enzyme metabolizing the drugs and glycoprotein. And then nitrile glycosides in the moringa, it is very important. So this nitrile glycosides in moringa, they reduce the metabolism of many antibiotics. So it would be very useful to give moringa olifera based product along with some of these antibiotics to enhance their bioavailability. Then Cuminium ciminum volatile oil component has got increasing activity on the efflux pump. Ginger has got powerful effect on the GIT mucous membrane. It increases the fluidity of the mucous membrane. It remarkably enhances the bioavailability of many synthetic and medicinal plant based products. And they've also studied a combination like Korean combination of 3 drugs in which Terminal cebula, Gingiva officinalis, and one of their plant has been involved. It was found to enhance the bioavailability of many drugs, especially those which are active in their treatment of the colitis. Apart from this, not much is known about how the intestinal bacterial flora handles the material which we consume. So intestinal flora contains trillions of bacteria which play a very important role in many of the physiological processes. In the GI tract, they play a very important role. Not only that, they even modulate the activity of CNS through their effect in the GI tract. So especially those drugs which contain glycoside bond are likely to be highly susceptible to the hydrolytic enzyme present in the intestinal flora. So for our product, it's also very important to determine the microbiome profile. So this can be done by using feces suspension obtained either from human or animals, and adding the drug and assessing after some time what are the metabolites that get generated in presence of feces suspension and in the absence of feces suspension. So this has been attempted for some of the products, especially nutraceutical products. Then the last component of the pharmacokinetic, distribution. So distribution studies are very difficult to study with complex formulation like our compound Ayurvedic formulations. If it is single moiety, it is possible by radio labeling it. It can be assessed how it get distributed in different organs of the body. However, in one of the studies, these Korean scientists, they have determined dehydrocastus lactone. Dehydrocastus lactone as the important active principle. To that, they have attached a flamma, a fluorescing material, and then they scanned it. So, as shown above, so it's shown. It has this figure depicts the accumulation of that dehydrocortisone moiety of the formulation and different places at different time intervals. So that's about the background information about the pharmacokinetics. Now coming to the methods which are available, or the parameters or the protocols available for us for pharmacokinetic studies. So they can be broadly categorized into those based on computational pharmacokinetics. It's purely based on using in silico approaches. The second one, which is the most important, which is the one which has to be used even after computational pharmacokinetic, is physiologically based models. That is the anatomy physiology based pharmacokinetic models. And then, of course, along with that, we need to factor in dynamic physiological blood tissue barriers, as I mentioned above. There is a model for assessing the effect on this blood and tissue barriers also. For intestinal absorption, 4 kinds of models are employed. In vitro models, in which certain kinds of cells are employed. Ex vivo models, in which a part of the organ intestine is taken out and kept in a experimental chamber. And then in situ perfusion model is there, in which small intestinal segments are perfused and used for assessment of absorption. And of course, in vivo models, they are given to intact animals like a rat, a dog, a monkey, or mini pigs to see how they get absorbed. Of course, this computerized model, compartment models, examples are CAT model, ACAT model, and ADAM model. These are softwares available, for assessing this probability of absorption through computational method. There is another important concept known as dispersion model. So whenever a drug enters bloodstream and from the bloodstream to the organ, the manner which it gets dispersed in an organ. So the dispersion of a drug in organs is not uniform. They say, at least in case of some drugs and material, the dispersion dimension, dispersion is different. To express that, they have coined a term known as dispersion number. They determine the dispersion number as part of pharmacokinetic profiling also. So for metabolism studies, the liver cells, cultured liver cells from primary hepatocytes, or those derived from cancer tumor of the liver, like HEPG2 cells, HUH7 cells, they are used. And microsomes are obtained from the hepatocytes after centrifugations, or we can employ precision cut liver slices to study drug metabolism. So in silico means computational methods, no animal is required. They can predict intestinal absorption before synthesis of a compound if we know the type of compound chemistry, or the chemical profile is available about a particular family. So this in silico method is not easily applicable to our product, because some of them may contain 1,000 active moieties. It's not possible to have information about all of them. And it will assess only passive type of absorption. It will not help us to assess the active processes. And it is of no use when complex interplay is there. Physicochemical method, it is just again depends upon the chemical nature and lipid solubility. The partition coefficient between octanol, which represents the lipid configuration, and water. So very simple. It is good for simple chemicals, but it has got very low predictive value for our compounds. Then artificial intestinal membranes are being used. So they have some role, and they may be useful with a different refinement for studying the absorptions. So this is the in silico model, which is not applicable to us. Just to show you the names: SIMGAP, Gastroplus, JSM, which I already mentioned to you. Then artificial membranes. There is one artificial membrane known as parallel PAMPA, that is parallel artificial membrane permeability assay. So when a large number of molecules are involved, so this can be employed as per high throughput, creating high throughput assay system. Again, it is only predictive for part of absorption process. The lipophilic compounds are retained on the membrane. The absorption depends upon the passage of the drug through the membrane, depends upon the lipid composition and pH of the membrane. Then we have got isolated intestinal cells like echo cells. So relatively easy to form. However, there is tremendous variability within the laboratory and between the laboratory, and we require radio-labeled material. And moreover, these intestinal cells they live only for 4 to 5 days, sometimes only for 30 minutes. So they will not be helpful for determining the effect of long-term administration. Everted intestinal rings or sacs, so they are quite useful, in which the human tissue obtained during operations or the intestinal segments obtained from animal, both can be used. So the only limitation is that non-specific binding may be there. Viability again is less than 30 minutes, and the steering conditions are suboptimal, which are not as like what is available in the intact intestine. So this is the experimental scheme for Pampa. So this golden yellow represents artificial membrane, which can be modified, which can be prepared as per our requirement by incorporating different material. Test compound is put in the apical chamber, and after some time, after different time period, the solution samples from this chamber is taken out, and the presence of active principles, a different kind of chemical, is assessed. Then Keiko cells. Keiko cells are derived from immune colorectal carcinoma. These cells, they've got a characteristic feature. Whenever if these cells are taken out and put on a semi-permeable membrane, so surprisingly they get arranged into a membrane similar to the intestinal cell membrane. So hence it is useful to study drug absorption. It is quite useful. So no bioanalysis is required. Only buffer solutions are sufficient. Transport mechanism in a limited manner, because they do not possess all the transporters. And we can add material which enhance the absorption and study the absorption enhancing material effect. You can evaluate the toxicity of a compound on the cells. And moreover, it is human origin, hence the species variation consideration will not be there. The limitation is that they lack mucosecreting cells. They do not have mucosecreting cells, even if they are present. They are present in a very minute fractions. So mucous layer is not present. So that's not natural. And the layer which forms on this cell is thicker than what is available in the intact intestine. And these cells are cancer cells. They are not normal cells. They have got different metabolic enzymes. They do not have all the metabolic enzymes, and the junction between the cells is quite tighter in this monolayer in comparison to the actual intestine. And as again, within the laboratory and between laboratory, variation is very high. Apart from the Keiko cells, dog kidney epithelial cells are used. Pig kidney epithelial cells are used. Rat fetal intestinal epithelial cells are also employed. And another group of Keiko cells in which the requisite enzymes get expressed or can be incorporated, can be used. Or we can use HT29 cell lines for co-culturing with Keiko cells. so that these cells produce mucus and simulate the normal configuration. So these models allow dynamic permeability measurement through monolayer. So they also permit detailed characterization of the biochemical mechanism involved in transport, such as involvement of receptor. But as I said earlier, by modulating the characteristic feature of the artificial membrane, so all these factors can be studied along with the measurement of expression level of messenger RNA and proteomics. So there is this experimental diagrammatic diagrammatic sketch of the experimental setup. As you can say, this is the transfer plate which is used for culturing the cacao cells. Cacao cells, when they are placed here, they form this layer. This is the apical chamber wherein the drug is put, and this is the basolateral chamber where it is conceptualized that the drug metabolite get accumulated. It's very important to see the integrity of the cacao cell monolayer formed. So that can be tested by measuring the transepithelial electrical resistance. If they form a proper layer, so they exhibit transepithelial electrical resistance. The electrical resistance gets exhibited when they form monolayer. So only those cacao cell monolayers which resistance exceeding 400 ohms per square centimeter are to be used for this purpose. Otherwise, monolayer formation is not complete. Again, that test has to be repeated. Ex vivo models can be used by using a apparatus known as Wishing chamber. I'll show you the diagram for Wishing chamber a little later on. And this is the setup for perfusion experiment. The intestine is the part of the intestine excised from the animal is is placed in the experimental chamber. Drug is placed here, and then these two openings are tied, and the loop is placed inside the chamber containing physiological salt solution. Krebs density to the solution which is present here, and it is aerated with carbogen to provide a condition which is similar to the natural conditions, and then periodically the drug containing in the intestinal interior is taken out and measured, assessed for different metabolites. A general metabolic drug metabolism scheme has been given. That is the first thing is chemical profiling and standardization of the product which we use, and then giving it to the human beings. From the human beings, metabolites are assessed by employing LCMS, MS, and other instrumental procedures, identifying the pharmacokinetic markers, and based on that to determine the pharmacokinetics. It is good for single components, but it is very difficult for multi-component testing. So different models for synthetic drugs, for example, for stability, liver preparation enzymes, metabolite identification, CYB inhibition, induction, transporters. So different kind of experimental processes have been mentioned here. So you can access this presentation through the organizers. So for drug metabolism, primary hepatocyte culture can be used. Primary hepatocytes got the advantage of being natural. So hepatocyte obtained from humans and animals can be used. So it is advantageous to study in different species, because if any species differentiation is there, it can be easily identified. It stimulates the natural condition very much. However, one of the major important limitation is that, again, the viability. They are viable only for few hours or few days at the best. The second one is epidermal cell lines. can also be used, but these cells do not express all the metabolizing enzymes. Only some are expressed. However, they can be used for preliminary studies. Microsomes, the hepatic cells can be centrifuged to obtain the microsome. Microsome contain both the rough and smooth surface of the endoplasmic reticulum. Thus, all the enzymes required for drug metabolism are present, including those which are present in the cytoplasm. Uh, and advantage is that since they do not have any cell membrane, there is no limitation. So there will be direct contact between the drug formulation and the drug metabolizing enzymes. So this is helpful for mechanistic study, though it is not natural to understand the mechanism or the involvement of different enzymes in metabolism can be easily assessed through this employing microsome. Precision cut liver slices are they provide excellent model for assessing biotransformation. So they almost maintain in vivo like conditions, including complex liver architecture, cell contact, cell polarity, and they're easy to prepare, and they can be prepared without using proteolytic enzymes. Liver slices from various species can be used to assess the species differentiation. However, the major drawback again is that the viability. They will survive only for 5 days, which limits their long-term studies. So NASHA is one of the important mode of drug administration and even a therapeutic measure in Ayurveda. So in vitro models are available to assess the effect of drugs on the nasal membrane. The nasal mucous membrane from animals like rabbit, sheep, bovine, and dogs is used. There is one tumor-based cell line known as RPMI cell line. So this cell line again they form a monolayer in chamber like housing chamber, the other chamber which I have mentioned earlier. So they can be used, but their availability is is a question. And they say that investigators who have used this cell line, they say that it's a good representative of nasal metabolism. And metabolite identification, there is chemical profiling, as probably mentioned by Ravindraji. So different instrumental facilities are employed for that. For pathway identification, through which pathway the chemical is metabolized, the inhibitors of the pathways are employed, and the co-administration of the inhibitors would give us an information about which pathways are probably involved. Like that, for P450 pathway, we have got aminobenzotriazole as the inhibitor. For MMO, we have got paragelin as the inhibitor. For FMO, we have got methimazole as the inhibitor. Like that, enzyme-specific inhibitors are there, which are used to which are employed for the study of identification of the major metabolic pathways involved in different this thing. Having said all and said that the in vitro procedures are important, but there are we have whatever result we get should be qualified by mentioning the limitations. So systemic effects are lacking in these in vitro systems. Chronic dosing is not possible. At this point I've already mentioned. Just to recapitulate them. Chronic low dosing may require multiple events that may not be obtained in the in vitro conditions. Theoretically, it is possible to maintain these in vitro models for long period, but practically it is impossible to maintain them for more than few days, because in long-term treatments require administration for months to years. So it is not possible to maintain the integrity of the in vitro models for such a long period. So that's why they can be ruled out for employment of long-term drug administration. So it's not possible to develop single in vitro system to measure all the parameters required. So because of that, recently another important development has taken place. So they have developed what is known as integrated discrete multiple organ co-culture. So this system has been employed for three type of studies to determine the cytotoxicity. So cell from kidney, cell from liver, cell from heart are cultured, and the drug is administered, and the overlaying. I'll show you the diagrammatic sketch explaining this. So we'll ensure the communication between different organ based cell cultures, and how the drug acts on those cells can be determined. For example, aflatoxin, when administered in this configuration, was found to specifically destroy epidocytes, not affecting renal and other type of cells. And also how the drugs get distributed between different organs can be simulated here by culturing the cells taken from different organs. And metabolism by multiple organ can also be determined here to some extent in this procedure. So this is the chamber. So in each of this chamber, there are three compartment, 3 or 4 or 5 as required. So in each of the compartment, different organ cells are cultured, liver cell, kidney cell, or cells derived from the other organs are cultured here. And this overlaying medium ensures that whatever developed here can get distributed with other chambers, so that inter-organ connectivity is established. So this has been proved to be quite good for single medicinal plant based products. For studying blood-brain effect on blood-brain barrier. So they have taken insect, the locust brain. The locust brain has got a specialized characteristic feature. It has got the enzymes for drug metabolism. It has got efflex system. It has got a flex system, and it has got anatomical structure simulating the blood-brain barrier. So, and it is easy to get the administered drug directly and see how it get metabolized, and also how it is handled. So this is one of the extensively used experimental ex vivo system for assessing the effect of drugs on blood organ barrier. So many computational systems are available. So this is this is a very brief explanation of the kind of experimental procedure available to us for carrying out pharmacokinetic profiling, but they are not quite suitable. Just to give you one or two examples. In one of the examples, the Chinese people have studied one of their compound classical formulation containing 4 ingredients. For this, they collected information about the chemicals, and they employed a computational method using computer to assess which of them are water soluble, which of them are lipid soluble, which of them can be candidate for transporter mediated transport, like that. So they have used this in silico method for total chemical components, and then they carried out in vivo studies in Sprague Dawley rats. Here it has to be noted, they have given the drug for 3 days, and 12 hours after the last dose, so they collected blood periodically from the orbital fluxes, and then for in vitro studies, they have used everted intestinal sac of the rat. So, and also they have used Keiko cell monolayer, the procedure which I had explained earlier, and they were able to obtain information, lot of information after the interacting of the drug with the material at different time intervals. Again, so this procedure has got some value, but it's of limited value. So for that, nowadays a new concept has been employed. This is the study carried out in Korea, South Korea, by Ki Lan and team. So they have coined the word polypharmacokinetics. So they have used different methods it is the metabolomic approach. So here, in vitro systems have not been used, only in vivo systems. So, they are given the drug to the human being, they are given the drug to the experimental animals. In human beings, they are given over a period of 6 weeks. And they have collected urine sample, blood sample, and they have kept two groups, one control group, and they have analyzed the urine and plasma of the control group people to determine the how many type drug related metabolites, that is the active principles like flavonoids, alkaloid, which may be present in any of the ingredients, whether they are present even without giving the drug. That basic level information they obtained by assessing the pre-dosing samples, and also dosing in the material or the samples obtained from the control group at various periods. And also they given the drug to the volunteers, and they collected it. And for this, they employed a LC-MS based system. And two important things are involved in this, in metabolomic approach. So the metabolic generation is characterized, but the main drawback is that highly sophisticated instrumentation is required to be employed. We need highly skilled people. Of course, they do not factor in the Ayurvedic concept of rasa panchaka, because we don't know what type of chemical configuration is responsible for imbuing rasa panchaka profile to a drug. Which are the chemical factors which are responsible, for example, this katu rasa, tirtha rasa, or madhura rasa, or different kind of vipaka. What are the chemical configuration which is linked to that? So these aspects have not yet been studied. So whenever we design any bioavailability or pharmacokinetic experimental procedure, it is very important to take those concepts into consideration. And another important feature is that employment of highly sophisticated statistical approach, multivariate statistical approach is employed to even before for pre-treatment methods. They say they employed what is known as deconvolution, alignment, integration. So the data informativity is maintained by processing by employing these pre-treatment methods. And they employ multivariate analytical techniques like principal component analysis, PCA, well-known statistical procedure. Then PLSDA. So many such statistical approaches are employed. So because here they are not handling 2 or 3 substances, they are handling 100, sometimes more than 100 metabolites or chemical configuration at a time. So for that, a simple statistical classical statistical approach would not be useful. For that, we have to apply system biology linked approaches in which this multivariate statistical analysis is employed. So they say that they have done this with one of their formulations, and they have studied all the aspects, and they also studied how these metabolites get linked to the endogenous, how they react with the endogenous molecules, and what happens after interaction with the endogenous molecules. So they have studied this in a formulation as pure rare T polyphenol's in humans, and as I mentioned here, they have employed highly sophisticated instrumental techniques for employing for identifying different configurations. So they were they say they obtained more than 6,000 features. And 176 altered variables from those. So another study has been reported by Chinese researcher on their compound preparation known as Kisanki dropping pills. Forgive me if I mispronounced them, because these Chinese names are very difficult to pronounce in a proper manner. So having said all these things, so I think the metabolomic based approach seems to be the comparatively better one for our purpose, but they have to be designed taking into consideration by linking the chemical configuration to Rasapanchaka profile and their bioavailability in the place where it is available. For us, this Srotas based approach is very important. So what are the component of different srotas? How much of these chemicals are available in that, in what quantity, in what manner, how they're configured? All these things have to be illustrated. That means it is not possible for a single group of scientists to work on this. So we require experts from different background, experts from Ayurveda, Dravaguna, basic principles, scientists or scientific workers from chemistry, molecular biology, pharmacology, biotech technology, omic, studies. Again, they come in molecular, biotechnology. So like that, by involving such a group, at least we have to prepare a general guidelines. At present, we do not have even a single, attempt to enter into this field. But it is going to be a must in the future, and it is going to be one of the most important and most studied, research studies in future. So this presentation, and along with the three important reference papers on which I have based this presentation, have been given to the organizers. If any of the participants want to go through them, please request them. They will send it to you. So with these words, I thank all of you for your kind attention, and also the organizer for providing me an opportunity to interact with you people. Thank you very much.
Thank you, Dr. Ravi Shankar sir, for a very informative session. Thank you.
I'm sorry,
Thanks
I think I've taken 1 hour
friends,
45 minutes.
Now we will move towards our third session. For that, we have an eminent speaker, Professor Dr. S.S. Saurika sir. Sir is known for his studious and straightforward attitude. Sir is former professor in Rasashastra Vaisheshya Kalpana at R.A. Podar Ayurveda Medical College, Mumbai. Sir is having 40 years of teaching experience. He is former Vice Chancellor of Gujarat Ayurveda University, Jamnagar. He was advisor Ayurveda to Government of India. Sir has been honored by Varadekar Award for Social Work and Research in Medicine, Sundarlal Joshi Award for Outstanding Performance in Administration in the field of Ayurveda. Sir is recipient of Excellence in Ayurveda National Award by Medscape India. He is the recipient of Vaidya D.V. Shende Rasashastra Award. Sir has been honored with Pranacharya degree by Sri Indraprasthiya Vaidya Sabha, Delhi. He is honored with Ayurveda Martand by All India Ayurveda Specialist Association, New Delhi, and late Hirendranath Gupta Memorial Award by East India Pharmaceutical Works Limited, Kolkata. Sir has been nominated as member of governing body CCRS, New Delhi, member of court Banaras Hindu University, Varanasi, member of Scientific Advisory Committee, IPGT and RA, Gujarat Ayurveda University, member of Management Council, Maharashtra University of Health Sciences, Nashik. Sir is currently working as Chairman Scientific Body, Pharmacopoeia Commission of Indian Medicine and Homeopaths. Presently is advisor to Honorable Sripadji Naik, Minister of Ayush, Government of India. I request Professor Dr. Saurika sir to conclude the session with his expert views. Dr. Saurika sir.
Good afternoon, everybody. Am I audible?
Yes, yes. Yes, sir.
I will share, please allow me to share my screen first. Can you see the screen?
Okay, sir. Right, sir.
Okay, fine. Okay, so good afternoon, everybody. Friends, since post-lunch session, you are listening to two very eminent speakers of their own field, one from the chemistry and the another from the pharmacology. Dr. Ravindra Singh from the chemistry side and Dr. Ravi Shankar from the pharmacology. Both are my very good friends, my old colleagues. They have been contributing in development of pharmaceutical standards and pharmacology in the field of Ayurveda since many years. Dr. Ravi Shankar was instrumental in, was very active in teaching also. He was, as you must have heard his biodata also. He was, I know him since my days in Jamnagar. So both of them have told you regarding one regarding the shelf life studies and the other on the bioavailability of the drugs. My friends, these things are very important. When you speak about the pharmacology, when you speak about the chemistry, these things are very important whenever you are talking about drugs. Drug pharmacology and drug chemistry, they are the most important part of therapeutic efficacy of the drug. You have to correlate the chemistry with the pharmacology, which ultimately lead to therapeutic efficacy of the drug. Unless you understand this link, you will not be in a position to understand the nature of the drug, the whole nature of the drug. This understanding facilitates us to use the drug appropriately at appropriate place. So these things are very much important. Of course, although these things are very new to us, particularly the chemistry and bioavailability, but when you speak of Ayurveda and these things put together, you have to take a different approach. When you speak about the uh, modern medicine drugs, these things are very easy to understand because the modern medicine drugs are single chemical moieties whose structure is very well understood. That's why it is very easy to correlate their structure and function. It is very easy to relate the site of their action, to relate their distribution, their excretion, their metabolism, etc., etc. Their ADME is very well understood. But that is not much easy as far as Ayurveda drugs are concerned. Another very important factor is that the use of drugs, the Ayurveda drugs, I am talking about the classics, the drugs, our formulations, the plants, their use is described on not on the basis of their ADMEs as it is as known today. They are described on a different basis. They have different basis of their use. I'm not saying they don't have an ADME. Everything which is being taken internally will have ADME. ADME is absorption, distribution, metabolism, and excretion. Every substance, whenever you eat, you take inside your body, it has to go to all these four stages. Ayurveda drugs are not an exception to it. But their ADME is not very differentiable. Because of their complex nature, it is not very easy to understand. Or I will say it is not understood at all. And they are not described, their actions are not described on the basis of these four things. Dr. Ravi Shankar was repeatedly speaking about the Rasapancha. Although he is a man from pharmacology, all his talk was principally based on Rasapancha. He was trying to correlate the pharmacology of the known pharmacology or the techniques used in pharmacology with the Rasapanchak, because he very well knows that the actions of the drugs, the therapeutic use of Ayurvedic drugs are described on the basis of their Rasapanchak. Rasapanchak, as you all know, Rasavira, Vipaka, Prabhav, and Guna. So, everything which we know about the pharmacology, it needs to be correlated with the Rasapanchak. Rather, I will say, we will have to use this knowledge to understand the Rasapanchak, to understand the karmas. Karmas are also involved in guna. So to understand the guna karma, you can't deviate the Rasapanchak and the other things. The things which modern medicine people are doing now, taking an Ayurvedic drugs and just trying to carry out their ADMEs directly without understanding the basic of their use. That is not expected, my friends, from you all. We all. We have to use the technology to understand the Rasapanchak or the Guna Karma. So, till now in the last two lectures, you have listened to bioavailability and the shelf life in the perspective of chemistry and the pharmacology. I will like to correlate these things to understand the Rasapanchak or the therapeutic efficacy effect as described in Ayurveda. So it is with respect to Ayurveda. That's why I have titled my presentation as Bioavailability and Shelf Life with reference to Ayurvedic drugs. Now, now I will put this availability vis-a-vis with Bhaishya 7 Kal. Because you know, bioavailability is considered with you know the definition of bioavailability. I will not go into those details. You already have listened to all those things. Till now, you must have understood that bioavailability is the drug which is going to be made available to every cell inside the body, right from its intake to till it reaches to the site of action. That is bioavailability. So the part which reaches to the site of activity is the bioavailable drug. So now here, which is the site of action? That is the $100 question that you have to understand. Has Ayurveda considered the site of action of drug? If we consider the action of the drugs, where is the site of the action of the drugs? Do we have different sites of action of the drug? If there are there, the drugs, action of the drugs are the intake of the drugs, the dose of the drug, the time of the drug administrations are prescribed on the basis of these the quantity of drugs available to these sites of actions. As far as pharmacology is concerned, modern pharmacology is concerned, it is the receptor site. It is the absorption through the GIT tract. It is the absorption from the site of administration of the drug that is considered as the distribution site. Whereas in case of Ayurveda, it is a totally different aspect. So don't forget this, accept how these drugs are acting. So we have to understand how the drugs are expected to act, and on the basis of the action take is described. So can we have to consider the bioavailability with reference to these things? The first thing which comes to my mind is Bhaishya 7 Kal. So if you consider Bhaishya 7 Kal vis-a-vis bioavailability, what do we find? What is the action of the drug? And how these Bhaishya 7 Kal, unfortunately, very few Vaidyas, very few practitioners they use these Bhajan Kal in their practice. I know, I have seen many people. They don't consider, they every Vaidyas, if you ask them, how do you practice, how do you give a drug, how do you administer the drug? At drug 3 times a day, one in the morning, to afternoon, and evening. So, whether this time of administration suits the time of 3 to 5. So let us just have a overlook of this Bhajan 7 Kal. Very important points I will like to bring to you, keeping in the mind the points you have considered or learned, things you have learned about the bioavailability. It's a good thing that my lecture is kept at the end after knowing the bioavailability and shelf life. I will be coming to shelf life in the later part of my lecture. So it is very clearly said that most of the times Vaishajam abhyavaret prabhate prayosho buddha kashayansh visheshena tatra veda sudarsha. Usually, even in the morning hours, the drugs in Ayurvedic classics or wherever we are using the drugs, usually, prayosho means usually. Most of the times, there are certain exceptions, but most of the times the drugs are administered in the morning hours. Visheshana kashayansha, but kashayas need to be given at different times. Kashaya considered Panchavida Kashaya, we need to consider that Bheda Sudarshata. That we will consider. Now, many times people ask me questions regarding the dose of the drug. The question asked is whether the dose of the drug which is prescribed in the classics, whether it is a one-time dose or it is a full day dose. The question is always asked in most of the conferences. This question is asked by the students and they don't get the satisfying answer. Because we don't consider the Bhajan 7 Kal whenever we are talking about the administration of the drug, the dose of the drug. My friends, this dose of the drug is prescribed on one side and Bhajan 7 Kal on the second side. Now the dose of prescribed is a dose of the drug. Now this is to be given depending on the Bhajan 7 Kal. That means what type of drug you are giving. See the Bhajan 7 Kalas, most of the Bhajan drugs are to be given as a one-time drug. So these are the drugs which are need to be, these are a one-time doses are there. Now, how many times it is to be repeated? It depends on the action of the drug. What type of action you are expecting from that drug. So this is a single time of drug, but at the same time, how many the time of repetition of the dose is dependent on the action of the drug. So there are Deyaha Panchavidaha Kala Bhaishyagrahane Nrinam. There are 5 broadly classified. Sharandhar has classified the Bhajan 7 Kala in the 5 times. As you know very well. So, Kincheta Suyodayo Jate Tatha Divasa Bhojane Sayantane Bhojane Cha Muhuschapi Tatha Nishi. Now, Prabhate Prayasho Buddha means at what time Prabhate? The Prabhata time is Kincheta Suryodayo Jate. That means as soon as the sun rises, just after the sunrise, you have to take the medicine. That is the Prabhata Kal. Another Kal is Divasa Bhojana. The morning hours of Bhojan. Divasa Bhojana is the time of Bhojan. Your lunching hour. The Bhojan. That time, the time of administration related to the Bhojana Kal, Divasa Bhojana Kal. The third is Sayam Bhojana. So one is related to Divasa Bhojana, second is related to Sayam Bhojana, third one. The fourth one is Muhur Muhur, repeatedly. and the last one is the Nishi, that's in the night. So these are the 5 broadly divided Bhajya 7 kalas. Now, if a drug is to be given for multiple times, you have to you give the same dose at multiple times. So this is a single time dose. It's not a full day dose. Remember very well. And that is why, but it can be repeated as the action of drug is proposed. Now how these kalas are given? This is very important, my friends. What it is said, now how do you decide the dosha and aushadha? Now drug action or drug administration, how it is decided, how the dose is decided, how it is decided. You have to consider all the 12 factors. You very well know it. I'm not going to repeat it. You are able to see the shloka. Dusham, desham, balam, kalam, maralam, prakriti, vayaha, satvam, sadhva, etc., etc. Dosha aushadha nirupanaihi. Sukshma saksham samikshesham, dosha aushadha nirupanaihi. So you have to relate the dosha and aushadha sambandha. Broadly speaking, you have to consider all these factors. But still, the 3 factors are most important. What is the relation of dosha and aushadha? What is the relation of agni and aushadha? And what is the relation of karma and aushadha? That means on which particular dosha the aushadha is required acting, on which type of agni the aushadha will be acting, and what type of karma you are expecting from the drug. If you understand these relations, you will understand at the time of administration of the drug. It is very important, my friends. But apart from that, it is very important to understand that these time of administration are to be considered in you are treating a patient who is not an emergency. It's a usual course of action. But if suppose there is an emergency is there, you don't have to consider the Bhajya 7 kalas, because it is very well described that atipateshu rogeshu na iched vidhim imam bishak. In case of emergency, you don't have to consider about vidhi. Forget about the vidhi. Forget about the time of research, etc. You have to immediately give the drug. Just like Pradeep Tagar vat shigram tattah kuryat pratikriyam. Just like there is some burning, house is burning, you don't look for time. No, it is not time of giving. Person is suffering, he needs immediate intervention, drug intervention. You have to give the drug without considering the time of administration, which is 7 card. But in a usual course of action, you have to consider the time of administration and the shot, all these actions, specifically the relation of drug with the dosha, with the agni, and with the karma, and of course the other things also. But 3 these things are important. Now this is very interesting. In short, I will be telling you exactly. Now, dosha is very important, my friends. So if you see, you can see here, though in doshas also, the most important is Vayu. Yeah, you know, Vayu is Balvana. Vata is Balvana. Because this control, Vayu is Tatra Yantra, Yantra Dhaha, it controls the other dosha. So you have to consider particularly the different types of Vayu need to be considered. In case of first, in the morning, Pitta and Kaphodreka is there, because Vayu doesn't come in the picture in the morning hours. But Pitta Kaphodreka, particularly for Virek and Imaman, for those 2 things, you have to give the drug in the morning hours. And of course, for the Lekhan also. So, Prayaha, Pitta Kaphodreke, Vireka Manarthi, Lekhanartham Bharisajam. This is the first time of administration. That is the morning hours. Whereas the second time which we have seen bhojan, that is, which is related to the morning lunch, morning meals. The second, pratap bhojan that we are considering. Here, different vayus are considered. What are those vayus? As you know, if there is problem, apana dushti is there, bhojanagre prashasyate. You have to consider the bhojanagre. That means you have to take the drug first and then you have to take the meal. Bhojanagre. You first take the drug and then you have, because it will act on apana. So it is known as apana kala. It is known as apana kala. Whereas in aruchi, etc., you have to mix the drug inside your meals and then. Whereas in samana vayu, what you will do in samana vayu mandegana agnideepan dadyat bhojana madhecha. In between the bhojan kal, in between the meals, you have to take the drug. First you start taking meals, start your meals. In between you stop, you take the drug, you again take the meals. So that is what is done with samana, because samano agni samipasthaha, as you know very well. Samana vayu is responsible for agni, stimulation of the agni. So if you want that there is some disturbance, samana vayu dushti is there, you have to use the midst of the meals you have to take. But then vyanagope bhojanante samahare. At the end of the meal, you have to take this. Whereas hikka akshava kampeshu puro ante bhojana, that is samudga. That means right from the first time before starting the meal as well at the end of the meal. At both the times, you have to take the meals. Whereas in udana vayu, we have taken the other vayu. In udana vayu, you can say here, grase grasantare deyam. It's very important. And this is to be given in Sandhya Bhojane. Udana Kal is the Sayam Kal. So in Sayam Bhojane, you have to give grase grasantare. That means in between the two morsels, you have to give it. In between two morsels, you have to give the drug. When? In the evening hours. Whereas in Prana Dushti, Sandhyasya Bhuktasya Amtecha Diyate. That means in the Sandhya Bhojan, at the end of the meals, you have to take the drug. So this is very important. Why vayu is important? It says, the Adamala has given the very beautiful commentary he gives. He says, Anilastu dosha dhatu maradinam pradhanan tamaha. It is a main dosha. Why? Because prakopa karana hetuscha. It is prakopa karana. The main cause of inflation is vayu. So ataha evasya prakopena pitta kapoyoho prakopa. When it gets inflated, it also inflates the other two doshas, pitta and kapha. That's therefore ekaha prakupito dosha sarvana eva prakope. Only one vayu is inflated, the other two doshas will also get propagated. That is why at the time for drug administration, you have to consider the vayu. And that too, different types of vayu you have to consider. And how you will identify? Depending on their actions. What is the action of prana? What is the action of udana? What is the action of vyana? What is the action of saman? And what is the action of other vayu also, apara vayu? So you have to take into consideration, accordingly, the type of function which is getting disturbed, you will understand which type of vayu is disturbed. And accordingly, you will understand the exact time of administration of the drug. So at that time, you might have understood here that except the samudga part, or except the two times bhutasya ante chadiyate, agre and ante, the both parts you have to give him. You don't have to repeat the dose of the drug at two times. Muhurmushcha, Trishna, Hikka, Chardi, Shwas, which requires continuous muhurmuhu drug needs to be given. Sun, or if sometimes, if not simple, a patient is not able to take independent drug, the drug can be mixed inside your meals and he can take it. Pachana, Shamana, and Lekhana, Bruhana, and Urdhaya through Vikar, you have to go Sayam Bhoj, Bhejanam Nishi. In the Vejanam Anannam Nishi, that too in the inside. Last time we have seen the Sayam Bhojane. But in this case, Lekhana Brahana and Udatukar, without taking any meals, he has to take the drug in the evening hours. That means if he has taken the drug, the meals, then sufficient time has to go before the stomach, when the stomach becomes completely empty. The Jirnanne, Jirne, that is the Ananna Kal. So in Ananne Bhejane, Anannam Bhejanam Nishi, the Ananna Kali, in the evening hours, you have to give the drug in the evening hours in Anannakal. On empty stomach it is used. So in short, it is very clear that in Apana Dushti, you have to give prior to meals. In Samana Dushti, in the midst of the meals. In Vyana Dushti, you have to give post meal in the morning. In Udana Dushti, post meal in the evening. Whereas Pran Dushti, in between the two morses, there is grasa antari. There is the in short area. So this why this is done, my friends? Why these particular kalas are important? Why these times are? Because when you give these drugs at this particular time, the drug will be absorbed appropriately and will act at appropriate place. The Apana Vayu, where it is acting, Apana Vayu is active in the lower part of the body. So, if the drug needs to be activated at the lower part, you have to take it before the meals. It will reach there and it will start acting, and the meal will follow that. So we are expecting that the drug should reach the that particular part. Site of action is the lower part of the body. It is not exactly the particular point, but it is the whole lower part, the Apana Pradesh, you can say. You have to consider the whole Apana Pradesh where the action of the drug is expected. So the drug will usually go. That means the drug will go through all these stages. Now, how it will reach, that will I will show you how it reaches over there. So this has to reach over there, and that part will start action. Whereas in Samanakal, you know, the Saman is active at the time of taking. Saman is in full action, in full swing when the digestion is at full go. When you are taking the meals, the Samana Vayu starts, it continuously stimulates your Jatra. So Samana Vayu, Samano Agni Samipasthaha. So it acts the drugs which we are expecting, which need to be, we expect their action on Jatra need to be given in Samana Kal, which is to be given in the midst of the meals. That means you have to take some meal, then in between he has to take the drug, and again he has to take the drug. So it is very necessary that a drug expecting the in Samana Agni, Samana Vayu, the actions of Samana, you know. That means if there is Agni Manja, you have to take the drug in Samana Vayu, Samana Kala. You are expecting Samana Vayu, Samana Kala. So that's why you are saying. In Vyana Kala, post meal in the morning. Very post meal, that too in the morning. Why? Because Vyano Ridhistitaha. And what is the action of the Vyana Vayu? Vyana Vayu distributes all the rasa throughout our body. So distribution of rasa throughout the body is expected from Vyana Vayu. So action of the Vyana Vayu starts when the rasa reaches the Hridaya. So from Hridaya it is distributed. So we are expecting the action of the drug in the Hridaya, Vyana Vayu. So before expecting action of a drug in the Hridaya, it has to be taken in Vyana Kal, that is in between the two post meal in the morning. So this, or even in the Pakshaghata, Vyana Vayu, because Vyana Vayu is all over the body. So these drugs are taken post meal in the morning. This is very important, my friends. So you have to take into consideration all these actions. Simply taking bioavailability, whether you are not considering every cell, no. These drugs, we are not considering a single molecule taking out from the drug and it will reach the receptor. It is not expected, my friends. Some action, of course, some of the phytochemicals may be acting on receptors. But here we are expecting the action of the whole drug. So they will act if you take because they are prescribed on the basis of these actions. They are not prescribed on the basis of their action on some receptor inside the body. Of course, they are prescribed on the basis of their action on Agni, Dhatu Agni, Bhuta Agni, Jatra Agni. Yes, but that is why you have to take into action, consider the site of Agni, Jatra Agni, Bhuta Agni, Dhatu Agni. There, the availability of the drug to the Dhatu Agni you can consider. Availability of the drug to the Bhuta Agni you can consider. Availability of the drug to the Jatra Agni you can consider. Specific Dhatu Agni, Rasa Agni, Mansa Agni, that you can consider definitely. So you have to consider the bioavailability with reference to the Dhatu, Bhuta and all these things. You don't have to consider to every cell. It will be different. For Asthi Agni, different type will be required. For Mausa Agni, different type will be required. So these things, and they need to be considered in reference to these Vayus. So which type of Vayu is responsible to take that drug to that particular Dhatu? It is very important, my friends. So these things you have to put whatever you have learned, you put these things to understand these things. Don't try to put these Vayu, this concept in that pharmacology. Don't try reversely. Unfortunately, the modern people are doing differently. They are seeing Prakriti in genomics and saying Ayurgenomics. There is nothing. You know, the word Ayura has no meaning. Ayur, everything. Ayurceuticals, Ayurgenomics. Ayur has no meaning. The word is Ayu. Ayurveda is a Sandhi. Ayuhu and Ved. When they join together, they become Ayurveda. Now, Ayurceuticals, if it is Sandhi, Ayuhu and Ceuticals. Now, what do you mean by Ceuticals? Ceuticals doesn't have any meaning. They may be pharmaceuticals also. They may be nutraceuticals also. So what you are talking about? Ayurgenomics. Ayuhu is life and genomics is different. So unnecessarily using fancy words, deforming the Ayurvedic principles, don't do it, my friends. And raise objections when such words are used. Raise your voice and try to stop spoiling of Ayurvedic principles. So try to use, I am not against the modern science. The science knowledge is very important, but that knowledge should be used to understand these principles. What we are doing? We are deviating from reforming these principles, destructuring these principles to suit them in the technology, that knowledge. So don't do that. So this is very important, my friends. Now, in this case, same thing. This I have already explained you, so I don't want to repeat the same thing again and again. Now, abhakta. It's very important, my friends. Why this? Why abhakta? Empty stomach. Now, empty stomach. Even modern science also understand. Abhakta is, that means when the stomach is empty, the absorption is very fast. Absorption is very fast. Now, when it is to be used? Empty stomach, when it will be given? It is to be given only when the patient is balavana. Pratah balavanam upujit. So empty stomach is in the early morning. So it is pratah balavana. Balavana. Tashmisha jirne punarahariti annannam. Ananna kala. Hestane anne jirne tadaushadam upujite. So whenever you are expecting such type of action from these things, then you can use it. But remember very well, if the drug is very tikshna aushadhi cannot be given empty stomach. Abhakta cannot be given, otherwise you will burn your stomach. There will be ulceration. Tikshna aushadhi, katu aushadhi, they cannot be given early in the morning, empty stomach. So this is very important. So tasmisha jirne punarariti anannam tacha niranna upayogad hetoho ativiryam bhavati. Because they are very active. Atitikshna. They will be very strongly acting. That's why the abhakta call, when you are expecting a strong action from any drug. But before that, you have to ensure the drug is not very irritating, not very tikshna. It cannot be given. That time, and the patient is balavan, in that case, you can give the abhakta call. There is a difference between abhakta and pragbhakta, remember very well. Pragbhakta and ananna. Pragbhakta and another different thing. Pragbhakta is just before taking the meals. That's when you are going to start the meal, and then you have, before that, you take first the drug and then start eating. So, itarastu pragbhaktadikam anna samsarganitam na atigrhanikaram bhavati. Anna samsarganituna na atigrhanikaram bhavati. The drugs which do not cause any glani, nausea, such drug can be given. So, it is very important that apana anila vikrita avadha kayasya baradhartham tadgateshu vadishu prashamanaya krishikaranartha yojjam. When such type of drugs are given, particularly emaciating drugs, lekhana dravya, krishikaranartha yojjam, when you are expecting to depletion of your body fat depletion, that time you can use the drug pragbhakta kala. Madhyabhaktam madhye bhaktasya tat samana anila vikrito voshtagateshu vajushu. So in some cases, madhya bhakta, in between the samana anila vikrito, that we have already considered. Adho bhakta also we have considered tatu vyana vikrito pratyarashat. This also we have considered. This also vyana pratyarvajanottar udana sayambhana. This we have already discussed. Now, a very important concept, very important point we have to consider is regarding vyavai. Vyavai, vikasi, etc. You all know the poisons, poisonous drugs, madhya, poisonous drugs, etc. These are very important. Now, bioavailability, we say these actions, these karmas, vyavai karma. What is vyavaitva? Vyavai, in short, is instant dispersion. As soon as the drug is taken engulfed, it immediately spreads inside. It gets distributed all over the body. Purvam vyapya akhila karyam tataha pakam bachyati. That means it is first distributed all over the body, then it goes to, it is digested. So vyavai, this is very important. That means there are certain drugs which are immediately distributed inside the body. That means they immediately become bioavailable. They are not digested. Usually, we know they have to go through the action, the digestive process. They are taken, first they are digested, rasa, then vipaka, and then then they start their actions. They have to get digested. But here, no digestion required. They are directly distributed inside throughout the body. So this is they're instantly distributed inside the body for their action. So if you are expecting some action, instant action, what you generally do, you tag these drugs with other drugs. This vyavaitva is we know that vyavaitva is associated with vishadravyas or madhya vishadravyas. This is actually. So what we do, we mix vishadravyas with these type of drugs, and then we give the drug. For example, bhanga, phenum, ahiphenum, etc. Shigram vyavai bhavad ashu apnoti kevalam deham shigratvad ashu apnoti. Vyavaitvad kevalam deham vyapnoti name vyavaitvam sarvatah prasana shirato patanaya patita tailavat. Just like they have given an example of oil. If an oil drop is put on a surface of the water, it immediately spreads on the water. Just like that, the vyavai dravya, if it is taken, it immediately spreads inside the body like anything. Another important karma is sukshmatva. It's very important. These drugs become immediately bioavailable to all the body cells. Sukshmatva, sukshmo guna vishesha, sroto anusarena shilatvat. Immediately it spreads throughout the srotasa. Dehasa sukshidresu vishesha ati sukshmuchyate. For example, Saindhava, Kshaudra is honey, rock salt, honey, nimba taila, eranda taila. Or in fact, all taila are like this, sukshma. Tailam, there is one statement, Vishasya tailasa natinchidantaram. In case of sukshma, they are all sukshma, tikshna, sukshma. That's why taila paka, siddha taila. If you want sukshma action, you can medicate the oils and then it can be given for their action. Sukshmatva, very important. In this case, in the light of this, one karma is very important, which is abhishandi, which obstructs the spread of the drug, which is obstructing the action of the drug, which is very important, my friends. This you need to understand. This action is very important. Unfortunately, this particular action is not understood. Now, what you have to do to understand this action exactly, you have to know the meaning of this action. What is the meaning? The abhishandi. The word abhishandi is very interesting. You break this word, abhi and syandana. Abhi is a prefix and syandan is the correct word. Now, what is syandan? Syandan is oozing. To understand anything, any Sanskrit meaning of the word, you have to exactly break it. Sandhi vigraha you should know. How that word is found is very important. Sanskrit words have definitive meanings. If you understand the meaning of the word, you will never forget their action and their use. So abhi and sandan, there are two words, different words. So abhi, you have to use the word whenever you have to give pressure on a particular action. For example, abhinandana. Abhiman, mana, abhiman. You know, there are many abhinandana, like that. Ananda, abhyanandana, like that. So abhi, if you catch that, you added a prefix, that means you are intensity of that action is shown. So the word is simple sandana. Since abhi is attached, it is progressively, intensively action. That means intensive oozing. Abhisandana is intensive oozing, in short, you can say. Intensive oozing. The intensity of the sandana, oozing is shown by this particular action. So because of that, these things happen. What happens because of this sandana? Of what sandana? What is extruded? What oozes out? What is the substance oozes? This is dosha dhatu manasrutasam kleda prapti jananam. What oozes out? And from where? From srotasas. It oozes out. What oozes out? Kleda. Kleda oozes. Now, what is this kleda? Kleda relates to every liquid inside the every fluid. So, in short, we can say serous exudation, we can say. Because serum is spread all over the body in every cell. So, serum oozing, that serous exudation, we can say excessive oozing of serum, excessive exudation of serous exudation is abhisandana. Very plain words. Just try to understand it. What happens because of that? Even every cell contains serous fluid, so it also exudes. The serous exudation is there, and because of that, the cell swells because of the exudation. It swells. Even the intracellular space also the serous gets inside the intracellular space, and because of that, the intracellular space also gets clogged. It gets blocked, and swelling of the cells, blocking of the intracellular space leads to the obstruction of the srotasa. And it goes to srotasa avarodha, particularly rudhva rasavaha siraha. It creates obstruction. So abhishandi is an obstruction of srotasa because of excessive serous exudation. So it is intensive serous exudation, excessive serous exudation, it because of which there is kapha vriddhi. That's why term abhishandi is described by different commentators differently. Of course, meaning is same. See, Dallan says, doshadhatu marasrotasam kleda prapti janam, or abhishandi upalepakari. Because of this excessive exudation, it gets saturated and kapha upalepa. Then Himadri said, abhishandi srotasravi. Fortunately, it is little mistake is there. There is srata. Actually, it is srotasravi. Srotas also starts secreting, exudating, serious exudation. Abhimukhyena syandayati. That means abhimukhyena. Principally, it starts exudation, serious exudation. And because of that, what happens? Malinyat srotasam srutirupam shleshmanam karoti ityarthaha. And because of that, the srotasa get coated by that excessive serous coating is there. After some time, the serous gets converted into thick mucus. Thick mucus. The mucus gets thickened and it coats the epithelial layer, surface, cutaneous surface of the internal srotasa, internal channels, and it gets blocked. So that's why in simple terms, if anybody asks us, what is it? Because many times the modern medicine people ask our people, what do you mean by abhishandi? When we say don't eat curd, because it is abhishandi, but they don't understand what is abhishandi. So when you are asked what is abhishandi, you are not able to answer. Tell them it augments the serous exudation. It augments serous exudation. They will understand. Now the example you can give, you tell them when you are having common cold, then if you eat this curd, what happens? The running of nose increases, continuously increases because the cough increases, because the exudation increases. That is what. So this is abhishandi, very important, my friends. So abhishandi, this action acts against bioavailability. Very important, my friend. This is very important. It blocks the channels. Now, Another important term is Yogavahi. Now, what is this Yogavahi? I'm very interested, my friends. This Yogavahi term is again mistaken. Many times it is not, we are not able to describe it appropriately. There are, very interestingly, it has been described by Dallana as well as Chakrapani also. You understand, you should understand, you remember this. Yag dravyena sanyojyate tadrak karma kurvanti. Yag dravyena, that means you, whatever drug it is associated, it will acquire the action of that drug, with whichever it is acquired. Yag dravya means with whom it is associated, it will acquire the action of that particular drug substance. So that is the Yogavahi. Or samana anukari dravya prabodhita shaktitvad iti bhavaha. That means it will help the other drug to enhance its own action. That is Yogavahi. So in simple terms, what happens, usually it enhances the bioavailability of the drug. Actually, it enhances the therapeutic efficacy. But how that efficacy is enhanced? Because it enhances the bioavailability of the drug. It enhances the availability of the drug to the body at the site of the action. So this is very important, my friends. So all Yogavahi, so how? Rasadravya, Rasaushani, Parada, Mercury, Yogavahi, Vishadravya, Yogavahi, Taila, Yogavahi, Grita, Yogavahi. Now, the Yogavahi action of these drugs are important, my friend. They are used in the drug. That is why all the drugs are tied with them. And then their actions are enhanced because of the Parade, the most important, most of the drug formulation. Why Parade is attached? Because in different formulations, different forms, its own forms, because it enhances the bioavailability. Its own action is not much important. It's a very, except in Rasindur, the compound acts. In other cases, the sulfur is more active rather than the mercury. And mercury has the Yogavahi activity. That is the most important role of mercury. Mercury is not absorbed, in fact. It's absorbed even if the global absorption is there. If you go take it continuously for hours, days together, it gets penetrated inside the mucosa and it may cause some problem. Irritation, injury, it will cause injury, or intestinal injury also. So this is Yogavahi. The ultimate effect is enhancement of bioavailability. Yogavahi cannot be translated as enhancement of bioavailability. Remember very well. I am telling you the effect of Yogavahi. Yogavahi is it enhances the action of the drug with which it is associated. That is most important. How? It is likely that it increases the bioavailability. Okay? Now, most important thing, my friends, in relation to bioavailability, again, these things are very much important. Unfortunately, we neglect. This is the most neglected subject of Ayurveda. And it is very unfortunate. It is very important, but you learn this subject, you study this subject in this first year in your Sharir Kriya Shastra and then forget it permanently. But this should never be forgotten. This is important for every physician. You need to understand this subject very carefully, because it tells you how the drugs, how the food material is transported to the expected site of action. Very important. There are 3 laws you know very well. 3 nyayas or 3 laws you can say. What are those laws or nyayas? They are Kedari Kulya Nyaya, Khale Kapot Nyaya, and Kshirdadhi Nyaya. These 3 nyayas will tell us how a drug substance, drug molecule reaches to the site of action. The site of action can be Dhatvagri, can be Jatragni, can be Bhutagri, can be anything, can be a particular organ, anything. Can be a single cell also. But that drug has to reach there. The same thing they are talking about this receptor site, receptor. So how does it reach? So this reaches by these 3 actions. They are very important. Never forget it. If you forget them, you will never understand the digestion and nutritional process, nourishment of our body. Very important, my friends. So same principles are applied. Usually this is described in the case of poshan, nutrition, dhatu poshanakam. But it is applicable, or it is in action to drug also. After digestion of the drug molecules are carried forward. How? First is the Kedari Kulya Nyaya. What is Kedari Kulya? Kedari Kulya, there are again two terms, Kedari and Kulya. What is Kedari? Kedari is water basin. Kulya is the channel. So the law governing law of water basin and irrigating canals. So you might have seen the fields. In the fields, the water is carried to the plant through the canals. There are water fields are there, water basins are there. Every plant is planted on a water basin. It has a small water basin, and one canal goes to that water basin. So the water goes to that water basin through the canal. So slowly it travels from the source of the water, from it may be a water pump or a well or whatever it is, or river. From there it travels, the water travels slowly, it reaches to the first plant, then it gives water to that, then it goes to another plant, then another plant, in line it goes away, like that. So just like that, here also the drug molecule will travel like that, and the plant which requires the body constituent which requires its own material, it will take up that material what is required. The material which is not required, it will not take, it will go ahead, just like that. The second part is Khale Kapot Nyaya. Khale Kapot Nyaya is pigeon and paddy fields. The law of pigeon and paddy fields. Pigeon is kabutar, kapot, and khale is paddy fields. So paddy fields, there are paddy fields. When the paddy fields are there, the pigeons come over there, they take the grains from the plants and take them in their beaks and go to their nests. Their nests may be nearer or far away also. So, but they directly go there. They nowhere stop. Just they take the grains in their mouth and they go to their nest where their children are waiting. Bird children are waiting. So this is like that. So just like that, here also there are certain ingredients which are directly taken from the digestive material, and they are directly taken to the expected site of action. This action may be, if there it may be quickly action also. It depends where it is recruited. The third one is the Kshiradadhi Nyaya. Very important again. This law is conversion of milk into curd. Immediately milk is converted into curd. So this is the metabolites. That means this is the class of the drug where the drug in itself is not acting, but its metabolites are coming into action. There are two types of drugs, remember very well. Some of the drugs are there which they act in their own forms without getting disturbed. They are absorbed directly and they go and act them directly. But in some cases, they are digested, their metabolites are produced, they are totally different from the basic drug, and these metabolites take the action. So in this case, this Kshiradadhi Nyaya is applicable, because Kshira is converted into Dadhi. That means the drug is converted into its drug metabolite. So metabolite conversion is very important. This is again quick acting drugs. So it depends what type of action. But these three things, nobody, I have heard so many presentations on the action of the drugs, but nobody has spoken on these nyayas in relation to action of the drugs, absorption of the drugs. These are the very important, friends, my friends. Remember very well. So you have to see what type of action you are expecting, what type of nyaya will be applicable to your own drug. So these things are need to be considered when you are talking about bioavailability, and try to understand the concept of bioavailability in the light of this thing. This whatever I am telling you. Okay? So now the next part, I will come to the next part, which is the shelf life. Please excuse me for 1 minute, because I will take some water, then I will talk. Another topic of today's discussion was shelf life. Now, this is again a neglected part. There is a general belief, I will say, misbelief. Actually, it is. People always tell that Ayurvedic drugs, however old they may be, they are very good. On the contrary, they should be as old as possible. I remember one day when I was working in one of the Ayurvedic colleges in Nanded. I wanted to purchase some powder, Trifala Churna or like that. So there was one medical shop which used to dispense the Ayurvedic drugs. So I went to him. He was a regular dispenser of our college people, patients also. So I just asked him, give me some Trifala Churna. So he gave me one packet. Then I asked him, how old it is, when it was put. He said, sir, it is old for ek saal ho gaya, sir. So I told him, no, I don't want this. I want just 2 months old, not more than 2 months old. And why? साहब ऐसा नहीं रहता, बोले जितना पुराना आयुर्वेद का दवाई है उतना अच्छा रहता है, बोले आप ये ले जाओ मुझे आपने. So I told him that I am teaching the same thing. I am a teacher. Don't tell me, my friends. So this is the general attitude of the people. And this attitude is very common in even educated people also, because our most educated people in our country are most uneducated in case of Ayurveda. If you take a survey, how many people of them, the people, educated people, very highly placed, even the IAS officers and engineers and all those people, bank managers, etc., you just go to them and ask them, what is Ayurveda? Few people will be able to answer. They don't know there is a graduation course is there and post-graduation courses are there, etc., etc., etc. So there is a lot of apathy is there. Anyway, I will not divert my point here. So regarding shelf life is very much important. The general guidelines regarding the shelf life of the drugs are described very well in the classics. It is known as Saviryata Avadhi. So Saviryata Avadhi vis-a-vis shelf life needs to be considered. Recently, Government of India, just 2 years or 3 years back, published a notification notifying the shelf lives of various formulations, that means various drug forms, not single formulation, not particular formulation, but drug forms, various drug forms, dosage forms, just like powders, churna, avaleha, asava, arishta, bhasma, kvatha, etc., etc. So just like that, the classics are, but they differ totally from the description in the classics. So I'll be concentrating on the classics. I'm not going to comment on that. So here, shelf life, you must have listened to lecture of Dr. Ravindra Singh, how it is calculated, accelerated studies, then real-time studies, various methods which are used, various methods of effect of packing on the shelf life, various types which are used. All these things you might have learned. He might have told you these things. But keeping aside all those things, how do you decide it? On which basis you decide that particular drug has crossed its shelf life? It is very important again. Usually structural disfiguration is considered when to decide the shelf life. So whether that is sufficient, let us see what our classics tell us and whether we can get some guideline regarding that. You learn that the modern techniques of deciding the shelf life, the methods of testing the shelf life, and all those things you have learned. Now, in that light, let us see this thing. Now, the terms are very much important, my friends. Charanda tells, Gunahinam bhaved varshat urdham tadrupam aushadam masatvyat tha churnam hina viratvam apnuyat हीनत्वं गुटिकालेहो लभेते वत्सरात् परं हीनः स्युः घृततैराद्याश्च चतुर्मासात् अधिकात् तथा औषध्यो लघुपाकः स्युः निर्वीर्या वत्सरात् परं पुराणः स्युः गुणयुक्ता आसवा धातोरसः. This is very important. And the first line, every term is important as far as shelf life is concerned, particularly the gunahinam. Now, what do you mean by gunahina? Now we have to understand that. This is very important, my friends, because unless we know the meaning of the term, we will understand how to test it. That means we have to see whether it is gunahina or not. So gunavina bhave varshad urdhvam. That means tad rupam aushadam. The nature of the drug will get disturbed after 1 year, after the lapse of 1 year. That's why the drug will not act. The nature, the appearance, rupa. Gunahinam bhavet swakaryam tad rupam yadrisham dravya swarupam tadrasham eva tad rupam. That means the rupa, the nature of the drug, whatever it was initially, that nature will be deformed after 1 year, after the lapse of 1 year. And because of that, it is not able to perform its own function. Swakaryam na bhavati ityarthaha. Gunahinam bhavet. It loses its action, it loses its characteristics, it loses its properties. Gunahinam bhavet swakaryakaram na bhavati ityarthaha. It is not able to perform its expected function, its expected action, because it loses its all properties, because it loses its nature. So what we have to examine after 1 year, or for testing shelf life, you have to examine its swarupa, its properties. You have to examine that. Swarupa is structure. You can see its microbiology. You can see its microscopic structure. You can see its content also, water soluble extract, alcohol soluble extracts. That you have to see. This is very important. This constitutes phytochemical history. You have to understand that. That will indicate whether it has become gunahina or not. So karyam, that if it doesn't have that, the nature, whatever are the indications which indicate that it is losing its nature, it's causing defective nature, it is directly related to its gunahinatva. Very important. So you don't have to go in very details. Even microscopic details will tell you that it is gunahina. It is losing its nature, its structure. So structural deformities will lead to its gunahinatva. That you have to see. But this is related to the kashtaushadhi. Dhatu rupam dravyavarjam. That means not for dhatu, because dhatu will not lose its nature after loss of 1 year, no. But plants will lose their nature after 1 year. So it is expected that the plants will lose their nature after loss of 1 year. Natu churna, not churnadi. This is not that means it is applicable for the drug as such, the raw drug. That means a plant cut plant is kept here, it will lose its potency after 1 year. But if it is powdered, natu churnadi kalpitam. If it is kalpana is done, if dosage form is converted into dosage form, it will not be stable for 1 year. Whole drug without any disturbance, it will be useful. It will be maintain its nature up to 1 year. Okay. Again, regarding churna, because churna doesn't maintain the masadvayadupari hina viryasya bhavaha hina virvatvam apnayat. Now, gunahina we have seen. Gunahina is related to the swakaryakaram nak bhavati. Gunahina indicate that it loses its functionality. It cannot function. That is gunahina, because of gunahina. But the same thing is not applicable for the hina virya. Hina virya is totally different from gunahina. Hina virya sabhavaha hina viryatvam apnayat. That means it becomes hina virya. Now here hina viryahani api atra bhavamatram natu sarvatha virarhitatvam. Hina viryatva doesn't mean that it has lost all its activity. No. It is bhavamatram. Some activity is lost, not total activity. That means those may be increased. It will have it. Virarahitam. Natu sarvatha virarahitatvam. It will not lose its total functionality. No. Whereas in gunahani, it will lose all its actions. But here, It will not lose its total action. It will maintain certain action, but the action will be slowly deteriorating. Nature is not deteriorating, but action is deteriorating. So this is very important. Virahitva, natu sarvatha virahitatvam. And that's why they have given the example of the multi-ingredient formulations. Multi-ingredient, for example, Lavana Bhaskarajina Bahuparanam Chirakala Sevanartham. The multi-ingredient formulations are used for a long time. If they lose their potency within 1 or 2 months, what is the use of preparing them? You will be at a loss. Etat sambharam bahukala upyogikam. If it is a multi-ingredient formulations, they will be useful for a long time. If they are not useful for a long time, annatha vyartham etat, otherwise such combinations are useless. They can be useful for a long time. Over a long time they can be useful. Tasmat bhav matram churnasya bayadvayat masadvayat upari hinatvam syaditi abhiprayaha. That means the hinaviryatvam is bhava matra. That means it will not lose its complete action. It will lose its action up to some extent. Now this needs to be examined. Now my friends, these are the dosage forms. They are talking in generalized terms. These generalized are general guidelines. In fact, these are useful for individual use. They are not for commercial use, which is very unfortunate. The industry uses them as a commercial venture for commercial use. No. In fact, each and every formulation has an individual shelf life. Individual shelf life is required. Every churna, Trifala churna will have a different shelf life, whereas Bhaskaravan churna will have a different shelf life. It's different. Bhaskaravan may be having more shelf life because it contains lavan. Sitoplazhi churna may be having a different shelf life. So these people, they have to test their shelf life continuously. Different product will have different shelf life. So they can't escape using the general notification, pointing the general notification. It is very wrong, in fact. I'm not in favor of general notification. It may be applicable for one, for example, some volatile contents are there. There are certain, for example, Tulsi. Now Tulsi has volatile content. Now how it can maintain its potency for 1 year? It may not. Vacha, same thing again. Cardamom, same thing. Lavang, same thing. It cannot maintain. The Lagudravya, they cannot maintain their potency for a long term. So they have to test each and every product individually for its shelf life. So they are very important, my friends. So again, same thing. Gutika, for Gutika, it has been said, for Gutika, Leha, Khanda, Avaleha, etc., etc., Chirakavasita. Api Swagunam na paritanje kinchit, kitu kinchit gunarahita. Kinchit gunarahita. I mean, these Avaleha, etc., they lose their action, little action they go on losing. It's not that they will lose completely. So that's why many times people say in the pharmacological circle, in doctor circle, they always say, even though it is an expired date drug is there, don't worry, only see that it is not deformed. You may take it, but remember that dose may be a little bit more. It will not cause you any harm. It will be guna, there will be guna, it will be kinchit guna rahita. Kinchit guna, it will lose some action, some guna. So this same is applicable here also. So Avaleha, Gutika, only thing you have to see that they are not infected, they are not deformed. It's very important. If there's some fungus is there, some deformations are there, then you have to throw it out. So you have to test it for that also. If some microbiological loads are there, then you can't use them. But if they are there, you can use it for a long hours also. So they have to test it. Just like that, Taila Ghritadyascha Chaturmasa Adhikat Tatha. Now this is again Taila and Ghrita. They are very important. Now what is this Chaturmasa Adhikat Vatsara? That means 1 year plus 4 months. That means 16 months. Taila and Ghrita, they are talking about the Siddha Taila, Sneha Paka. Sneha Paka are good, maintain their shelf life for 16 months. Shodasha Masad Uparitartha. Some of the commentators, some of the experts, they say that they are active, their shelf life is only 4 months. But when they say for 4 months, they mean to say 4 months of rainy season only. eke chaturmas grahane varsha kalam manyante. Varshayam ushitam sneham pakvam hinando apnaya. That means as soon as the avaleha crosses the period of chaturmas of varsha kalam, the monsoon period is over, 4 months of monsoon are over, they become useless. Their shelf life is lost. That is what the quotation says. Again, the same thing here also, they have to test their shelf life. Atra api bhava matram, very important again. That means bhava matram, that means only you have to take the sense that they are losing, they are become virya hani. That means they are losing certain action. Their dose may be more. Kintu, another very important thing, they talk about the snehapak, then avaleha, gutika, etc. Because these things, these formulations, they are having dravya samuha. That means they are multi-ingredient formulations. Because of they are gelled well in these formulations. Their gelling is there, and their gelling creates certain actions, certain properties. Now, since gelling gets disturbed because of over the lapse of time. So, dravya samuha pakajanya gunasya hani. That means it is very likely that over a period of time, this gelling may get disturbed, and the action which is obtained because of this dravya samuha, thus gelling, you may lose that action. Individual action may be maintained, but the action acquired due to gelling, their combination action may be lost. So, dravya samuha pakajanya gunasya hani, natu prakriti guna hani. That means their nature may not be disturbed, individual nature may not be disturbed, individual nature of the avaleha or taila may not be disturbed over the period of time. That means whatever the period they have taken, the chatra 16 months, but after that, the formulation will lose its combined effect, and it may be important, very important. So you have to consider that. So this dravya samuha pakajanya gunasya hani. This is Adamalla commentary, very important commentary. Usually we don't read such commentaries, but they are very important. Leha siddho gudadinam bhavite. So all these formulations, avaleha, sneha pak, guda, pak, etc., this principle is applicable to all these things. So very important again. Another thing regarding laghupaki aushadha. Laghupaka aushadho varshad uppari nirviryaha. Remember where here they are not saying hinavirya, they are saying nirviryaha. That means they will lose all their potency, complete potency. So nisheshena virya rahita ityarthaha nato hinatvam. Very important. Previously they were talking about hinavirya. Now they are saying nirvirya. Laghupaki aushadhi, they will lose their potency virya after lapse of 1 year. Varshad upari nirvirya syu nisheshena virya rahita ityartha natu hinatvam tasya yava godhum pramukha. In that they are telling, just like yava and godhum is wheat, barley and wheat, they will not be useful after the lapse of 1 year. They will be only useful up to 1 year. So they will become nirvirya, they are useless. Don't use them. Eteshu eva godhuma tilamasha navahitaha. That's why these things are useful when they are fresh. Rudha purana virasa tatha natata gunakarina. Purana is vatsaro ushitam. That means after 1 year. Purana relates to 1 year. After lapse of they have become Purana. So now virasa, after the lapse of 1 year, they become useless, nirvirya. So yava, tila, godhum, masha, they are useless. They become hinavirya, not hinavirya, nirvirya after the lapse of 1 year. So don't use them. But generally, other grains, other cereals, they maintain they are useful after the lapse of 1 year. Other grains, cereals, except these things, the yava, godhum, tila, masha, other cereal grains, they are useful after the lapse of 1 year. For example, Shukadhanyam, Shamidhanyam, Samatitam Prashastate. Samatitam is Varshatitam, after the lapse of 1 year. All Shukadhanyas, all Shamidhanyas, except these things, Yavagodhumatilam Asha, they are useful. They will become most useful after the lapse of 1 year. Purano Praya. Why? Because Purano Prayasho Ruksham. They become Ruksha Prayana Abhiram. When they are fresh, they are Guru and Abhishandi. So they cannot be used when they are fresh. Namam dhanyam abhishandi rahu samvatsara oshitam shiga janma tatha supyam nistusham yukti bharitam. Very important, my friends. They are talking about these real grains. They are whenever they are fresh, they are abhishandi. Lagu is samvatsara. Lagu means they become very lagu when they after the lapse of 1 year. Shigra janma, that means whichever material, the grains which are shigra janmi, that means they are ready for harvesting within short time. They are lagu. And supyam. Supyam means soup, soup which are tomato soup, etc., dali soup. They are the grains which are used for soup, preparation of soup. They are known as supya. Nistusham. That means after remove dehusked grains. And yukti bhajitam, and the roasted grains. They are lagu in nature. All these things. The grains which are usually cultivated and harvested within a short time, within 6 months, like that, they are very lagu in nature. That is why Shashtik Shali. What is Shashtik Shali? You know, Shashtik Shali is very important, my friends. You have to understand the meaning of these things. Shashtik means 60. Now that means the Shali, that is the rice, which is harvested within 60 days of its cultivation. 60 days after cultivation is ready for harvesting, collection within 60 days after cultivation is known as Shashtik Shali. It is very good, it is said to be, because it is very lagu in nature. So that is why Shigar Janma, it is known as Shigar Janma. So this is very important. Another very important quotation is Navani Yojjani Dravyani Akhal Karmasu. Vina Vidanga Stabhyam Gudadhanya. Usually every dravya should be used very freshly collected, except there are certain drugs which are used to be used very old, Purana. Purana means after 1 year old. There is Vidanga, Krishna is Pipali, Piper longum, and Guda, Dhanya, which we are discussing. Ajja is Grita, and Makshik is Shaudra is Madhu, honey. So these need to be used after 1 year. I am cautioning you as far as Pipali is concerned. In the market, the Pipali, fresh Pipali is available. Nobody sells 1 year old Pipali, my friends. Usually it is 3 months old, 4 months old. They crush. You can see it. If you crush the Pipali, the powder becomes a little bit greenish tinges there because it is very fresh one. So what you do, you purchase it, store it for 1 year, and then powder it. So 1 year old Pipali. Same thing is regarding Vidanga also. So recently we have conducted one study on fresh Pipali and old Pipali. Lot of differences there. We are going to publish the paper on that recently. One of my PhD students is working on that. So this same thing is on Vidanga also. Already paper has been published. So be careful whenever you are using. Ghritam abdat param pakvam hi niviram prayapte. Usually the Grita is the ghee. Usually 1 year old ghee is not good for eating, for drinking, for application of it. Tailam pakvam va chirasthai. But tail, it may be pakwa or apakwa, it can be used even chirasthai gunadikam. But at the same time, I told you the dravya samuha pakajanya gunahani, very important. That you know. Puranam rukshadi guna sayukta, that I. Another one thing important is shukshkam navinam yad dravyam yojjam sakala karmasu adram ja dvigunam yujjal. Now, for example, usually the drugs are used when they are dry. But if suppose you are having some fresh drugs, you don't have shushka dravya, you have fresh drugs. They have to be used double the dose, in the double dose. For example, the dry dose is suppose 1 gram. The fresh dose will be 2 grams, like that. So this is your Yujjadev Sarvatranashya. But there are certain a class of drugs where you don't have to use fresh one, but don't use them in double the quantity. Such drugs are Guduchi, Kutujo, Vasa, Kushmandisha. There are a number of drugs. Shushkaram Navina Yojam Dravya Sakala Paramsu. Sadaiva Ardra, Viguna Naiva. These drugs need to be always used fresh. They should not be used dry. Guduchi, Kutujo, Vasa. but you need not use them in double the quantity because they are not usually dry, So whatever quantity dose you want to, they single dose. They need to be doubled. Those need not to be doubled, but they need to be every time they should be fresh dose, fresh drug should be used. Okay. So that's all, my friends. In the light of what you have listened to the two lectures, I just wanted to throw light on the Ayurvedic perspective of these two: bioavailability and the shelf life part. I know that because I had already seen their presentation. This part might not have been covered by them. That's why I have covered this. So thank you for giving me a patient listening, and thanks the organizer for giving me a chance to address this gang thing. I think I must be speaking for more than hours, for 1 hour. So thank you very much. Over to Shishir. Over to the organizers. Okay, fine.
Thank you so much, Saurika sir, for this concluding session with your very nice and informative presentation. Now it's time to present a vote of thanks for today's webinar. I would like to thank our speakers, Dr. B. Ravishankar sir, Dr. Ravindra Singh sir, and Dr. S.S. Savrikar sir for enlightening us with their knowledge and interesting things. It gave deep insight into the topic and also revealed some interesting facts about Ayurveda. I'm pretty sure the precious knowledge of today's webinar will definitely help us. Once again, thank you so much for being with us. Thank you, sir. I want to thank President Dr. Ambadas Kulkarni sir, Secretary Dr. Abhay Kulkarni sir, Chairman of College Committee Dr. Abhay Sukhatme sir, and all other members of governing body of Ayurved Seva Sangha and Principal Ayurved Mahavidyalaya Dr. S.L. Dasri sir for encouraging us to organize this unique webinar session. I would like to thank the whole webinar team for their support in conduction of this online webinar. Also, thanks to all the staff members who worked hard to make this webinar successful. And in the end, I would like to thank all the participants in this webinar for paying attention and learning. Thank you. Thank you, sir.
Thank you. Thank you, everybody. Thank you, Ravi Shankar. Thank you, Ravindra.
Sir, thank you very much. In my next presentation, I will